And greetings. Welcome to the Evidence Reasons Academy. This is the sister channel to the main channel, Evidence and Reasons for the Christian Faith. I just got word that on StreamYard that Rebekah has to unmute herself. I don't have the power. I don't have the superpowers to unmute her. So Rebekah, please feel free.
Oh, sorry about that.
Oh, quite all right. Quite all right. So Godzilla Freak is here. This is just incredible. I can't believe all the people that are following this channel. So this is a nerd night, and in professional research, they have something called journal clubbing. So this is the way nerds do clubbing. So I call this Nerds Go Clubbing, and we're going clubbing on a Saturday night. I'm going to be talking about RNA. So Rebekah, I've made you an honorary nerd. So why don't you introduce yourself to the...
I'm not a real nerd.
Oh,
I'm not a real nerd, Sal.
Uh-oh, I'm in trouble now. Well, okay. If you make it, yes, if you make it through this evening, you're a real nerd. How about that?
Okay, cool. All right,
So,
I want to be in the nerd club.
Yeah.
Sal is my creation advisor. My name's Rebekah. I have a channel called Bread of Life, and I also have one called Examining Evolution. And I'm so thankful for the help that Sal has given me with that channel. He's done several streams on there and been a wonderful blessing to me. So thank you, Sal.
One thing I can do for you, I think you're going to be the first moderator on this channel, or at least one of the first.
Nice.
So can you type something in the side chat, and then I will give you your moderator privileges.
Sure.
And that way I can also put your information when you type it in for your websites, your YouTube channels, and I'll put that in the video description right now. Okay, so
great.
First,
thank you.
Let me see if I can do this right. All right, so... And I just did add moderator. So go ahead and type your Bread of Life channel and your Examining Evolution channel. I will then add it to the video description. So everyone,
here's Examining Evolution.
Did it let you add it? I didn't see it.
Yes.
Oh, I guess my system's just a tad slow. Oh well. Yeah. I don't know. I don't see it. In any case, we'll just ignore that little glitch and just move forward. If I remember, I'll definitely add it. So with that, tonight, the goal is to read this paper and to give some context. I have to tell you, this week was just wonderful. I got to talk to Fazell Rana yesterday, and I said, do you have any words for our audience or Christians out there who are also science students of science? And Godzilla Freak asks, are we waiting on Otangelo and Emory? Emory may not be able to make it. I don't know where Otangelo is, and we're just going to plow forward. And it's going to be more fun because, unlike Emory, who's a graduate student, Rebekah is a student of all sorts of things other than graduate level biology. So this should be fun, as we're going to be actually reading a graduate level, PhD level paper. And it's okay. You can do it in biology. Other disciplines, you'd have a hard time, but you can do it for a lot of things in biology. You just have to use Google and Wikipedia, and you'll get through it. So I was encouraged when Fazell Rana appeared, and I said, could you share anything for the students out there of science who are also Christians? He said, well, oh, sure. He said he was not raised in a Christian home, and he was an agnostic. He went off to school. He was in graduate school, his PhD program in biochemistry and chemistry. He was studying cell membranes. And he said, as he studied biology and the question of abiogenesis, he came to the conclusion there had to be a creator. And that led to his conversion to Christianity. I'll mention that someone named Matt Bilyeu on Rebekah's channel said he studied cell membranes. He was an atheist just before he entered freshman year college, studied biology, had evolution shoved down his throat. But senior year, fourth year of biology, he's studying cell membranes. He said only a creator could make this. And he came to that conclusion on his own, just from the science. So there are two people then that have trusted in Jesus Christ as Lord and Savior. And a big part of that was studying cell membranes. So this is what the Evidence Reasons Academy is about. I think that some of the fluff and the philosophical arguments, they may work for some, but for others, it's this hard-nosed scientific study that just plows over evolution using other disciplines of science that are far more respected than evolutionary biology. As we study that, I think it will give people the assurance there is a creator if God is working in their heart and they're impartial. And so this is why I feel it's important to give nerd talks, because a lot of science students are doubting Thomases, like myself included. And it's in looking at science and studying the right science that will enable us to see the evidence of the creator. I want to really point out, to this day, I don't know anyone that studied human evolution from a creationist standpoint and ended up being a creationist or Christian thereafter. Honestly, I can't name one. I'm not saying that they don't exist. And it's for that reason I tend to de-emphasize the question of things like what Erica would study, which is human evolution. And there's a debate among creationists, and it seems like a lot of Christians are so fascinated about, can you prove that we didn't evolve from ape-like creatures? And I don't go to that territory. You start with a cell, you start with the cell membrane, you start with molecular biology. And I have found more people, in my experience, have said that they weren't believers, and then they studied biochemistry and cellular biology. I haven't met one that had studied human evolution and said, humans were definitely created. And that's just kind of my persistent feeling. If you have any thoughts before we move on, Rebekah?
Well, that's interesting, Sal. It sounds like you're saying that the evidence for human evolution is fairly good. Is that kind of what you're saying?
I'm saying it's hard to refute. It's hard to refute. I wouldn't. They can make a stronger case, a very strong case for it, and I would have a hard time refuting it. So I focus then on abiogenesis, because if you can believe that you see, the thing is one will give the impression you totally lost the debate about creation evolution, but there's so much more to this. If you start off with abiogenesis, then it becomes possible to believe in the creator. The question then is, how did God intervene? Was it just at the very first life, or was it, say, also including the creation of humans? For a lot of people, if God at least made the first life, there is at least a God. And that's a big, that's going from night to day. So for a famous atheist like Antony Flew, it was the problem of abiogenesis. He was a famous atheist, and then he rejected it all based on philosophical arguments, and then also the intelligent design of the first life. So once you can accept one miracle, it becomes more feasible to accept other miracles. If you try to just start off with the ape I mean, the chimp-human split and ancestry, it's very hard to demonstrate a miracle unless you're really, really good. And even then, we don't quite have the sophistication yet to make just like a killer argument relative to things like abiogenesis. So we're going to go in between tonight, and it's not going to be exactly abiogenesis. It'll be kind of attacking human evolution a little bit from a different angle, and it's still important because then this would lend credence to at least some form of creationism for, like, not just the first life, but say the organisms that are higher on the chain of complexity, and that helps. So even Fazal Rana, who believes in an old Earth, he thinks that there are miracles of special creation along the way. So there are some similarities between his views and the young Earth creationist views. So today we're going to explore this whole question of junk DNA. So the question will be, why are we going to talk about RNA and biological phase separation? How does that help us believe in Jesus Christ? By the way, glory and honor to our Lord and Savior Jesus Christ. This is why this stream is here. It may not seem brutally evident that that's the reason we're doing this, but that's why we're going to suffer through this, is to give evidence for the Christian faith through the study of RNA and biological phase separations. The evolutionary biologists, the top tier, who specialize in what's known as population genetics, had realized if the human genome had a lot of complexity, that is, it had a lot of features. So you could, by way of analogy, we had very, very simple computers a long time ago, and they got increasingly more complex and more capable, had more versatility, more capability. And the evolutionary biologist realized if the human genome had a lot of functionality and lots of capability, it would also have a lot of places to break down. So the more, just like a very complicated car, it has just more things that can go wrong with it. Cars that didn't have radios and air conditioners, for example, those they wouldn't break on the older cars because they didn't exist on the older cars. So by way of analogy, if humans are really, really complex and have a lot of capability, there are just more places it can break down. And with the given mutation rate and the fact that most mutations cause damage, the evolutionary biologists realized that for evolution to be true, more than 90% of the genome roughly had to be junk. And this is well summarized by Dan Grauer's response to the ENCODE Consortium. The ENCODE Consortium had done research mostly to try to understand the function of the genome. Yeah, bless you, Rebekah. Don't worry, we didn't hear you.
Thank you.
The function of the genome and its role, because they're very interested in curing heritable diseases and also diseases like cancer. And so they're like, we're finding a lot of things related to cancer that we can identify in the genome. And they said, the evolutionary biologist told us that that's junk. We're having a problem with that because 90% of the heritable diseases or cellular, diseases that are related to the genome, 90% are in the areas they say are junk. And they came to the conclusion at least 80% of the human genome has function. And some evolutionary biologists like Dan Grauer and Larry Moran, biochemist actually, but he's an evolutionary promoter, and even others like Steve Matheson, who's a cellular biologist, they were livid. But especially Dan Grauer, he started calling the ENCODE researchers at the NIH, who happened to spend on the order of $500 million of research to further medical advancement, he started calling them crooks and ignoramuses. He was livid. And because they said, they dared to say most of the genome is functional. And that was intolerable because this looks like, they prefer to say this is just bad design, and 90% is just like the result of accidents. The idea that it's functional has kind of like, even on an intuitive level, suggests very clever and careful design. But then on another level, the population genetics number, they couldn't make the numbers work with that level of functionality. And in response to the ENCODE's proclamation, Dan Grauer said, if ENCODE is right, evolution is wrong. If ENCODE is right, evolution is wrong. So how does this relate to tonight's topic? Well, I was in a debate with Dr. Dan 2 years ago. It was a very cordial, civil debate, very intellectual. It was nerd heaven for a lot of people. It probably flew over the heads of most. And he was identifying all these regions of DNA that he thought were junk. And I said, Dan, I don't think it is. And I don't think I think it's too early to say. And then in other shows, I said, Dan, you guys are putting yourself out on a limb there saying it's junk. It's only a matter of some discoveries by the cellular biologists in the medical profession and biotech, and they start to find out it's functional. You guys are going to be toast. It's going to be embarrassing. Why are you sticking your necks out? And the answer is Dan Grauer, and the other Dan Grauer, respected evolutionary biologist. He said, if ENCODE is right, evolution is wrong. Do you have any thoughts here, Rebekah?
I'm amazed. I've never heard this before.
Okay, so
I've heard the discovery that, hey, this junk DNA wasn't, it wasn't as non-functional and junk as we thought it was. But I haven't heard this thing about Dan Grauer. Is that his name? Yeah.
Right. And so there's been kind of
interesting. Mm-hmm.
So you had a show recently on why ERVs are functional. And so they're probably functional on so many levels. I could point out some there too. So this is on your, you're welcome, by the way, you're welcome to show this on your Examining Evolution channel and upload it there. This video.
Thank you.
It takes about a day for YouTube to process it, where you could actually download it. It's a little quirk in their system. So just be patient and you'll be able to download it. But so what's tonight about? It's to really just call out the evolutionary biologist and said, you screwed up again, guys, big time. I mean, one of your own guys, 10 years ago, 2012, at a big meeting, and he's being cheered by all the evolutionary biologists. And he was just railing on the ENCODE NIH project and saying, if ENCODE is right, evolution is wrong. They're a bunch of crooks and ignoramuses. And the leader of them, Ewan Burney, is the scientific equivalent of Saddam Hussein. I mean, this is just terrible stuff he was saying. And I'm like, gee, you'd think he was criticizing creationists. There was so much vitriol. And he was really upset. And I was just like, the ENCODE Consortium had people from like Harvard, probably like MIT, Stanford, lots of the best universities on the planet. They had like 200 to 400 researchers working on the project, trying to solve human disease. And Dan Grauer is going to call them a bunch of ignoramuses. And like, dude, you got a lot of audacity. You got a lot of gall to say that. And so this is dedicated to people like Dan Grauer. This is just payback, because he said it's junk. And I said, it's just only a matter of time before the cellular biologists, they find something, you're going to have egg all over your face. So this is what it is tonight. So anyway, I'm glad you picked up on something and why this junk DNA fight is important. And the evolutionary biologists, as they continue to lose this, would rather forget about that episode. But I'm here to help remind them of yet another
How kind of you, Sal.
Oh yeah, I'm such a sweetheart. Yeah. On an earlier show, I was pointing out that the synonymous mutations end up mostly to be detrimental, and that's just blowing out thousands of paper. I mean, that was stuff I think even Erica or Dr. Dan were talking about. It's like, yeah, synonymous mutations are neutral. We're able to measure the strength of selection with that. And I'm just like, no, that's out of the water now. And that was just a paper like in the last few months, I believe. And I'm just like, yeah, okay, so this is really embarrassing, guys. I mean, can you get one thing right? I mean, seriously, can you get one major thing right? I mean, it's just bad. So, okay, so given that you're here,
Peter has a question in the chat.
Oh, why don't you read it to me while I pull something up? So
Okay.
You're going to help me multitask here, Rebekah. Thank you.
Yeah. So Peter says, generally, do you think vestigial non-functional structures or sequences really help the evolutionist argument? Surely even in a creation model, organisms have had time to lose functionality.
Okay, so physical structures are like, say, arms, limbs, whatever. They're much easier to knock out because it takes so much to get right to make a functioning organ. It's very hard to tell whether an individual gene has been severely compromised just by looking at the molecular sequences and doing cellular biology. And it's even at the organismal level and at the level of organs and systems, it's not necessarily easy to even isolate what specific molecular changes caused something to lose, an organ to disappear. So, for example, if the Hox cluster were mutated in a certain way, and we've done this experimentally, the fly's legs could end up on its head. And those are just a few changes. Now, we happen to know that that's what caused it because we were the guys, the scientists were the ones tinkering, and they actually put the mutations there and said, hey, look what we did. But if it happens in the wild, it's not quite the same. It's going to be difficult to reverse engineer that. So the question again is, do you think vestigial non-functional structures or sequences really help the evolutionist argument? I don't know. I put it this way. I wouldn't attack that. So the evolutionists will give you all these lists of why evolution is true. What creationists do, and I think they're so mistaken, is they'll try to address all the evolutionists point by point. And I'm like, that's kind of not good, because then you're fighting them on ground of their choosing. What you need to do is fight them on ground of your choosing. So rather than try to address their points, and they'll say you're not addressing their points, it's like, well, I'll grant you all those points. But how about the evolutionary eukaryotic components? How about the evolution of major protein families? Does anything of, even if we assumed everything you said about vestigial organs and all this other stuff, will that solve these problems? Because if it doesn't, evolutionary theory has serious problems, because these are way bigger problems. Wouldn't you agree? If you can't evolve major protein families, evolutionary theory is dead in the water. So what about similarity of human chimp ancestry? So what about vestigial structures? The issue is evolving complexity. So that's how I attack it. And all I've seen is these, the evolutionists will come out with a new headline and the creationists will just scramble to try to, spin it a little bit as best as they can. And like, guys, you're not experts in this. Don't try. I mean, we just tend to be reflexive to try to respond to anything they say. And I said, no, you take the initiative and you put stuff on the table yourselves. And the way you do it is you say, let's accept that for the sake of argument for now. But I'll point out this doesn't solve all these other problems. And that's the way to attack it. And they'll be silent and they'll say, oh, yeah, that is a problem. And I felt for Rebekah when she was talking to Erica, and Erica was showing her all these things about human chimp ancestry. I'm just like, oh, Erica, if I were there, this would be a different exchange. I'd be saying, Erica, that's excellent work on your part. And let's just say human evolution's true. You got to tell me, I'm sure you'd agree, Erica, that this wouldn't solve the problem of eukaryotic components, would it? Or this wouldn't solve the problem of evolution of major new protein families, like say topoisomerase or helicase, or I could just list a whole bunch. It wouldn't explain the problem of membrane-bound organelles or the evolution of translocation processes, and I could just go on and on and on and on. You see, that's kind of the way to turn it around. And then what I see the creationists do, and I just cringe. They'll say, "Oh well, we're actually just we're not exactly similar. There's 90% similarity. How do you account for the 10%?" And I just cringe. It's like, this is not the way to fight this battle. So Paleo Logo says we need to be proactive, not solely reactive. Absolutely. And Peter, Paleologos, I totally get it that this is your field since you're studying human evolution. And I don't know if you were here earlier when I was saying, I actually don't, this is, I appreciate your research. So there is your personal research area, and you'll bless other creationists. But as far as evangelism, for those who are like, say, friendly seekers, not the psycho scoffers that you see on the internet, but people genuinely interested or Christians on the fence, I emphasize cellular biology, the question of abiogenesis, particularly of the abiogenesis, the problem of membrane origins, cell membrane origins. At least 2 people accepted Jesus Christ as Lord and Savior because they studied cell membranes. So just a data point for your edification. And I don't mean to minimize your excellent work in the study of human evolution. It's great stuff. And, I have my specialized work that probably won't reach a lot of souls, like in heavy electron quasi-particles, maybe hardly anyone except a few people are interested in. So Paleologo says, I'm more interested in creating a creation model than debunking evolution honestly. And that is the view of a lot of professional creationist researchers. So we're kind of divided into the model builders. We're trying to reconstruct history and make a model of kind of the chronology, what are the created kinds, and they call it discontinuities. And then we have the anti-evolutionists like me and Dr. Sanford. The reason anti-evolution is close to the heart of me is that's what got me back to the Christian faith. So it's always nice to prove what is wrong. I'd like to know what is true. Well,
this is why I just adore Peter. This is why I love him. And this is the same heart that I think you have, Sal. And this is why, I just really have the greatest respect for you and for Peter. You guys really pursue the truth, and it's just so precious. And so I like Peter and Sal, once again, I just have to say, I feel like you guys are some of the most important people on the planet. And so thank you for the work that you're doing and for being a voice. And it's really beautiful to see that. And it means a lot to me.
So, there's a saying, you can't build your own house by burning down your neighbor's. I'm in the mode of burning down the neighbor's house. Okay. So Peter is in the mode of building. I'm having too much fun burning down the evolutionary house. And we all have our roles in the body of Christ. And so tonight is about burning down someone else's house. Okay. So Peter, if this isn't your thing, I take no offense. So I was just laying out why this is important for people like me. By the way, I was a pyromaniac growing up. It got me in trouble. The one thing that worried my parents was their son Salvador's backyard chemistry experiments. I was hospitalized when one of them exploded. I had shrapnel in my chest. I was hospitalized for 2 days so they could pull it out. So, yeah, I'm definitely a pyromaniac, literally and figuratively. I ended up joining, working for the Defense Department and just working on systems that go boom, and that's satisfied a certain personal curiosity. So, so now we've laid out what we're talking about today, and that is if ENCODE is right, that is if the genome is mostly functional, then evolution is wrong. So let me pull up a and we'll just start from the basics and then go from there. So, do you remember, Rebekah, the central dogma of molecular biology? Does that ring a bell? You won't have to state it, but does it? If it doesn't, I'll just
I've
share
heard that. Yeah.
So the central dogma of molecular biology can be pictured here. And on the left is DNA, and DNA is used to make, serves as a blueprint for RNA, and RNA then serves as a blueprint to make proteins. Normally we just do the shorthand and say DNA is a blueprint for proteins, and that is correct. But mechanically speaking, there is an intermediate step where the DNA is used to make a blueprint of RNAs, which then serves as a blueprint for proteins. So you could say it either way. This one is just a little bit lower in the weeds as far as details. And this description of this information flow is called the central dogma of molecular biology. So if you study molecular biology, at least with Dr. James Tan, one of the first classes, they'll say, this is what it's all about. And the rest of the class will then be talking about all the ways that all the mechanisms of how this is accomplished. Do you have any comments here, Rebekah?
No.
Okay, so what we're focusing on then, if the DNA is mostly junk, then one would presume that RNA is junk, that it doesn't have function. It just floats out there. And ENCODE realized that a lot of the RNAs that are supposedly junk are being transcribed, but they don't make any proteins. And so the evolutionary biologists will say, see, this is victory for our hypothesis because all this RNA is just made and it doesn't do anything because according to the central dogma of molecular biology, the RNA generally is going to be translated to make proteins. There are exceptions, like I think the tRNAs or something. There are a few others. But they said most of it, by and large, is just junk. And they're saying, we've been trying to study this for 30, 40 years and no one's finding function. You guys are crazy. And Larry Moran, one of Dan Grauer's fanboys, he's a professor of biochemistry at University of Toronto. He's saying this is junk. You're just wasting your money. We should have found it out by now. And people like CRISPR-Cas on the internet said, yeah, if it were functional, it should have been so obvious by now. And I'm just like, dude, it could be our instrumentation isn't that good. Our capabilities, maybe it's going to be discovered as functional once we improve our ability to detect things and figure out what's going on. Because certainly, and he was on my show and I didn't have a chance to tell him, I said, we wouldn't have known the DNA even existed unless we had all the laboratory capabilities. You can't just say because you can't see it functioning, it's non-functional. I said, that's ridiculous. I was saying that to myself. I didn't say it to him to his face. I tried to respect him since he was a guest on my show. But now that he's not here, I'll talk behind his back. So, all right. So that's the central dogma of molecular biology. And Let me go to another screen. And we're just going to go through the abstract of this. And let's see, we're already at 38 minutes. And there's no way we're going to get through this whole paper in one sitting. So just tell me when you start to get your eyes start to roll too much, Rebekah, and then we'll just call it a night.
Hey, I thought it was going to be a torture session.
Don't
let me off the hook too easily. You're supposed to be torturing me.
Okay.
And you said if I got through it, I got nerd status. So come on, we have to get through it.
All right. Okay, so let me tell you why this is of value to you. Some people will just say you're not sophisticated or whatever. You haven't studied this. In this one sitting, you're actually going to know more. You actually would have studied more than most of my critics. The guys who make shows about me or whatever and just say, I don't know anything. You could say, well, Sal walked me through this. And that looked really persuasive, that it has function. Do you have any responses to that? Do you want to contradict what's here in peer-reviewed literature? I mean, we'll read through this, and you tell me if that sounds like RNA, this supposed junk RNA, has function or not. And they're going to have to tell you with a straight face, oh, that's not proven. You know, we need more evidence. And I'm just like, where have I heard that before? I mean, that's kind of a double standard, because I'll demand evidence for eukaryotic component evolution, and they don't have any yet. They believe it. And I said, there's more evidence that this has function than eukaryotic components evolved naturally. Isn't that right? Because here it is, right in the paper. It's going to go in brutal detail how we figured this out. And we'll go from there. So this is the benefit to you. You can claim that you got your battle stripes here tonight. They'll say, have you read the peer-reviewed literature? You could say, I've definitely read at least one, and you can cite this video, and you could cite these papers. And I want to see how they respond to this, because as I was reading through some of this, I was hearing like crickets from the other side. Now, I know, Rebekah, you spy on some of these other channels that talk about me. If you find anything substantive, just let me know. I hope you don't feel I'm breaking your confidence, but sometimes when you report it back, you just laugh. I said, did you find anything? And you just laugh. And I'm like, okay, that kind of answers my question. So.
Yeah, you're totally blowing my cover here, Sal.
Oh, boy. Oh, I'm in trouble now. So. Okay, so I will also show you how to. I'll show you how to read through this, and it's learnable. It just takes a little perseverance. So the title is The Role of RNA in Biological Phase Separations. The role of RNA in biological phase separations. So what's a phase separation? Let's just begin with that. When you put oil in the water, you know how the oil will separate out from even if you stir it, it kind of separates out. So an emulsion is where you have two things that really don't mix, but you kind of force it to mix. And then you have there are all these chemical names for that. So phase separation means the RNA will start to separate out spontaneously. And it's dependent on the sequence. So DNA has the ACTG, and RNA has the counterpart of that, ACUG, where the T is translated to uracil, the thymine to uracil. That's neither here nor there. But suffice to say, each of the RNAs have their spelling. And it enables them, their sequence, depending on the sequence, it's going to relocate in a different position on the cell, which is just amazing. It's like a postal code. Now, there are lots of repeats, and I'm not going to get into that necessarily tonight. But those repeats are actually really important also in the organization if we start dealing with stuff like heterochromatin. This is way beyond that, but I'm just trying to explain the word phase separation. So just think when it says phase separation, it's meaning in different parts of the cell, based on just kind of like how water separates from, by analogy, how oil separate from water. Things will just spontaneously compartmentalize. So think about spontaneous compartmentalization. Isn't that really cool? That they just have different spellings and it's just going to kind of make a building spontaneously. You think of the mind that's able to figure out those sequences to get it to position itself. And this is what this is about.
Amazing.
It says here, phase transitions that alter the physical state of ribonucleoprotein particles contribute to the spatial and temporal organization of the densely packed intracellular environment. This allows cells to organize biologically coupled processes as well as respond to environmental stimuli. RNA plays a key role in phase separation events that modulate various aspects of RNA metabolism. Here we review the role that RNA plays in ribonucleoprotein phase separations. So it says RNA plays a key role. And I'm like, that doesn't sound like junk to me. Whatever the RNAs are, and I could cite more literature. So if you got nothing else from that, you could start out with RNA plays a key role. And so now we'll talk about some of the other words in here. And so we're going to painfully go through some of this. So remember when I started working for Dr. Sanford, I had no formal biology training. I had to take courses while I was working for him. So how did I read articles like this where I had all these terms that I didn't know? Well, we could thank God for Google because it can help us. So we have this word. Okay, so we could let's look at phase transitions. Let's just do a Wikipedia article. Sometimes you'll just not really get a good explanation. This isn't the most helpful. Sometimes you'll find out a term that's commonly used is used in this slightly different way in biology literature, and this might be the case. So it says, in chemistry, thermodynamics, and many other fields, phase transition or phase change are the physical process of transition between a state of a medium identified by some parameters and another one with different values of the parameters. Commonly, the term is used to refer to changes among basic states of matter, such as solid, liquid, gas, as well as plasma in rare cases. So we could see water goes from like the solid phase to liquid phase, and then the gaseous phase when it's steam. Is there any problem there? So RNA must have all these
problem.
I mean, is that sort of comprehensible what a phase transition is when it's changing from one state to another? Okay, so yes. All right, there we go. So that's how I read through it.
All we've done so far is the abstract. Let's get to the meat.
You know, Rebekah, see, you always want to get to the meat. I'm trying to set it up. We have to go through this methodically. This is how a nerd would go through it, okay?
So this is more like nerd training. You're training me how to be a nerd.
Right. So then we have this word called, and I'll show it to you now, the ribonucleoprotein. Ribonucleoprotein. So let's look up ribonucleoprotein. The reason I'm actually doing this, so I do this off the top of my head. I want to kind of be orthodox here, at least keep the textbook stuff. So let's go to the Wikipedia article. And it has nice pictures here. And unfortunately, this is kind of slow. By the way, I have another computer hooked up because this one tends to overheat, just blank out. If we do, just give me about a minute. I'll get the other one started up. It's this secondary Salvador computer here. Let me show you. It's easy. There, see him. So in case we blank out, we'll be able to recover quickly. So let's see what a ribonucleoprotein is. A ribonucleoprotein, an RNP, is a complex of a ribonucleic acid and RNA binding protein. So ribonucleic acid, that the acronym for that is RNA, ribonucleic acid. So an RNP is basically an RNA plus a protein. So when it says RNA binding protein, that means it's a protein that binds to the RNA. Bind means connect or attach. They use the word bind. I would have preferred they use the word connect. So let's see some pictures. See if they have any pictures of RNA binding protein. See if they have any. Any? Okay. Those are nucleoproteins. I want to see ribonucleic. I think they have one. Do they have one? I don't see any. Well, I don't have one off the top of my head. A picture of it. They do have a picture here. Let's see. Cell nucleus with DNA in blue and the nucleoprotein in red. I'll just forgo that for now. So just think of an RNA combined with a protein. So we got through the half the first sentence. Phase transitions that alter the physical state of ribonucleoprotein particles contribute to the spatial and temporal organization of the densely packed intracellular environment. So spatial just means kind of the positioning. And when it says temporal organization, that means just over time because the cell is constantly changing. And so it's just saying that the position of these particles depending on the time. This allows cells to organize biologically coupled processes as well as respond to environmental stimuli. RNA plays a key role in phase separation events that modulate various aspects of RNA metabolism. So because, I mean, this is just kind of logical. If RNA is part of the ribonucleic particle, then RNA has to be a key role in this. And this abstract is just describing how this article will go through here. RNA plays a key role in phase separation events that modulate various aspects of RNA metabolism. So when it says modulate, that's just varying it, changing what it does. So this is just like changing the mode of operation when it says modulate. Here we review the role that RNA plays in ribonucleoprotein phase separations. I think we're cool here. And I love pictures. So this is the cell, and you can see the edge of the cell here, the cell membrane, and then we have the nucleus here. So this must be what kind of cell is this? Prokaryote or eukaryote?
Eukaryote.
Oh, bravo! You're my favorite student. Because it has a nuclear wall, and you can see all the different kinds of RNAs that are made here. So we have these mRNAs, which make proteins, and then we have these other things called like poly-A mRNAs that create stress granules, and it's also used for other things. We have these rRNAs and these snow RNAs that are in here. These link RNAs, A to I, that's adenosine to inosine edited RNAs. That's a whole nother ones. Link RNA like MALAT1, and there are really lots of others. And then these cagal bodies, which I'm actually not familiar with. So I'm learning something here. So, okay, what's the meaning of this? And evolution is wrong, because they said if ENCODE is right, evolution is wrong. This is already starting to show the function of some of these RNAs that were not encoding and may have been presumed junk. So let's keep reading. Organization of the densely packed intracellular environment requires compartmentalization. This is particularly important for gene expression, as coordinated processes must occur in an ordered fashion. In eukaryotic cells, double-stranded DNA, dsDNA, is sequestered in the nucleus and packaged in histones. Within the nucleus, DNA is organized into heterochromatin and euchromatin to control the relative access to the transcriptional machinery. Transcribed mRNA undergoes splicing, adenylation, and capping prior to export to the cytoplasm. Each of these processes is spatiotemporal control that ensures correct processing and localization. So we had a show on evolution of eukaryotic components, and I was starting to get so in the weeds. You said, Sal, this doesn't, it seems so far away from the evolution of eukaryotic components. And I said, well, I realized it could seem that way because it took so long to describe. And so one of the terms here is heterochromatin and euchromatin. Does that ring a bell from our show on?
Yeah, because you said chromatin is the thing that's in eukaryotic cells that's not in prokaryotic cells that allows them to have all these different expressions that, you know, aren't expressed in prokaryotic cells.
You're good.
Thank you.
And I don't know if you remember, I think
You're breaking up, Sal. It's hard to hear you. Your computer is breaking up a lot. The last couple of things you've said have been very choppy, but maybe it's on my end. I don't know.
No, it's on my end. Whenever I search for something, I might have you.
Okay, let me search for the things. I'll search for the things. You tell me what to search, and I'll search it, and I'll read the
search
definition.
for chromatin.
Okay.
Search for chromatin.
Okay.
And just show up a nice picture of chromatin, and then I'll put yours up. And while you're doing that, I'm going to turn off the air conditioner because I'm starting to freeze.
Okay.
Okay, so
All
I'm going to
go
ahead and share it.
I'll share my screen. Okay.
Yep.
Let's see. Okay.
Lovely. So
you want me to read it?
If you want to, I was hoping you could find a picture.
No. Yeah. There's the picture.
Okay,
let me see if I can make it bigger.
Yeah. Okay, so you see the word histone. So you see the DNA is wrapped around the histone. So you could see the DNA, and it's wrapped around the histone. Histone's like a spool. And when you have a histone DNA around it, we call that thing a nucleosome. So now you're learning the terms, and then the all of that is called chromatin. That's generally unique to eukaryotes. Some archaea have which is a prokaryote, have some things that look a little bit like chromatin, but the way it's implemented in eukaryotes is spectacular. By the way, I don't know if you remember, I said I had a picture of the God-made random access memory and the man-made, and how they look very similar, with kind of all the beads on a string. So one way we can describe this chromatin is beads on a string, where the beads are the histones and the string is the DNA. So it's kind of like a necklace. And it just happens to look a little bit like some of the man-made computer memory of long ago, like the abacus and also magnetic core memory. So why did I bring that up? Well, let's go back to, and by the way, thank you for doing this, because then this is going to go a lot smoother, thanks to you. So
you're welcome.
Let me.
I stop share. So
you can, or thank you.
Okay.
No, that's fine, because now you helped me by doing that. Let's go back to here. This is particularly important for gene expression, as coordinated process must occur in an ordered fashion. In eukaryotic cells, double-stranded dsDNA is sequestered in the nucleus and packaged in histones. So now you just saw the picture of how DNA is packaged with a histone. Within the nucleus, DNA is organized into heterochromatin and euchromatin to control relative access to the transcriptional machinery. So there are two kinds of chromatin. Heterochromatin means it's really, really well packed. Like you pack it so hard that it's like kind of packing your bag, and it's to get to something you have to unpack everything to get to it. So just think of heterochromatin as really tightly packed chromatin, and euchromatin is the kind of chromatin that is unpacked enough that you can actually work with it and use it. So that's just the terminology. So this is setting up how RNA is going to deal with all this. So stop me if there's something that I didn't explain well. I will probably tend to over-explain it more than you need, but just stop.
Yeah, you can go on.
Okay, so transcribed mRNA. So do you know what mRNA is? It's called messenger RNA. So mRNA is the RNA that is used as a template for making proteins. It's it goes into a machine called the ribosome, and then the ribosome will read the mRNA, and then it'll make proteins with that from that blueprint. So that's what mRNA is. So you'll see a lot of things that will say like mRNA, miRNA, link RNA. There are all these varieties of different kinds of RNA. So mRNA is the RNA that codes for proteins. There are probably some subtle.
Okay.
That's probably some simplification. Someone will probably ding me on that, but that's close enough for our purposes. Transcribed mRNA undergoes splicing. I've never heard it stated that way. It's usually called raw RNA, but anyway, mRNA undergoes splicing, polyadenylation, and that means it's just kind of putting a little tail on it that's made of A's, adenine. So the RNA genetic code is adenine, cytosine, uracil, guanine, and the adenine, you're just adding a lot of adenine bases at the tail of this string of RNA, and it says, and capping, that means it puts something else. So you put a cap and then you put a tail on your message, a cap and a tail, prior to export to the cytoplasm. So all this RNA transcription is going on inside the nucleus, and then it's going to put this tail, and it's going to put a cap and a tail, and then now it'll be shuttled through the nuclear pore complex. So do you remember the pictures of the import-export through the nuclear pore complex? Okay, so this is great. This is great. You totally understand that.
It's coming together. It's coming together, right?
Right. You see, this is you don't need...
I mean, like every little piece that you're teaching me is starting to come together.
Right.
But when I hear this, I'm just listening to your description, And I'm thinking, how can anyone not believe that God is behind this, or a mind and intelligence? Like, come on. Like, it
Well,
has all the marks.
You interact with those kind all the time on your channel, and they seem to really want to avoid these details. Do you notice they'll change the subject if you try to throw this out on them, and when I try to assert it, they just don't want to go there. They want to talk about philosophy or anything else except actual physical facts like this. And this is why I enjoy just talking about it, because I notice when I'm on your channel, they don't want to talk about things I want to talk about. I said, okay, I get it. They say, what's your evidence for God? And then I'll get cut off when I just I said, I could try to be succinct. And I said, evolutionary theories of failure. And they'll just, attack me from that point. I'll never be able to go in this much detail. It's called Brandolini's law. A little bit of BS takes an hour to refute. So a minute of BS takes an hour to refute. See how long it's taken just to get to this point. But then if people are willing to spend the time, they'll see it. So I'm glad you're seeing this now.
I need to. Oh, I mean, to me, it's evident even without looking at, as zoomed in on a cell or on DNA and RNA and all. It's evident just from observations in life,
Right.
So I feel like anyone, even before we had this kind of information, I think the evidence for God is there in the created things. So,
and I think though, at this level, it starts to become harder and harder to deny. In Darwin's time, it was easy, but as the years progressed, it got increasingly difficult. Now it's starting to reach really the breaking point for a lot of people, and thank God for that, because we had one of them on your channel, Matt Bilyeu, and then we had one on my channel, a PhD in chemistry, biochemistry. He worked for Fortune 500 companies. That was Fuzz Rana. And his work, along with Hugh Ross, converted a Nobel Prize winner in chemistry. I mean, this would have been unheard of.
Who was that?
Richard Smalley. He accepted Jesus Christ as Lord and Savior as a result of things like this, or it at least reinforced his conversion to Christianity. And yeah, it's limitless. We can keep going on, but going back to the actual article here is, so now we're seeing the role of RNA. It's setting up what's actually going to be controlled by the RNA. So this is only the second paragraph in the paper. Each of these processes is under spatiotemporal control that ensures correct processing and localization. It has to be in the right place at the right time. I mean, I like those very nice academic words because it sounds, it seems to have gravity to it. But if I were to use kind of more pedestrian language, it's basically saying, this makes sure it's at the right place at the right time. So they use the phrase, spatial-temporal control. Spatiotemporal control. I mean, to me, I kind of like that flowery language, but anyway. Just as membrane-enclosed organelles, and we've talked about that's a feature of eukaryotes, and that's very difficult for evolution to explain. Just as membrane-enclosed organelles, nuclei, mitochondria, endoplasmic reticulum, Golgi apparatus, serve to organize biological processes into discrete cellular domains. By the way, you have such a nice voice. Would you like to read some of this instead of me?
Sure.
Why don't you give it a shot?
Why not?
Okay, let's get it out. Okay, here we go.
Okay, wait, where are we? Okay, so non-membrane enclosed where we are? Non-membrane enclosed domains play a similar role in the organization of biological activities throughout the cell. These are defined by the physical nature of their constituents and are referred to as membraneless organelles due to their ability to concentrate factors associated with biological processes, typically involving RNA metabolism. MLOs serve to concentrate their components in a way that facilitates processes such as transcription and ribosomal RNA or processing in the nucleolus, or sequestration, such as translation initiation, complexes, and signaling molecules in stress granules. MLOs can be visualized using phase contrast, immunofluorescence, or bright field microscopy. The lack of a limiting membrane allows MLO constituents to rapidly exchange with the surrounding environment, allowing a dynamic response to changes in the cell state. Wow, there's a lot of stuff in there. It kind of lost me.
But you sounded so sophisticated, Rebekah. Oh my goodness. You're going to blow away your critics. I mean, honestly, if they keep telling you you don't understand, it's like, okay, can I read this to you? And why don't you explain it to me and explain why this evolved? You could practically go through this whole paper and say, okay, I don't understand this. Can you explain it? So they'll say, you don't understand how evolution works. And I'll say, okay, so why don't you first explain how all this actually works in the present? Now explain how it evolves. And of course, you're going to get brushed off in a variety of ways. And so you go back to school and learn it. Like, that's what they'll usually say. But you can call their bluff. I did. I actually did go back to school. But let's go through this.
Okay.
So we have the membrane enclosed organelles like the nucleus. And I just learned something. Okay. They have these things called membraneless organelles, these MLOs. So we have MLOs. So these things, so a membrane is made of what? It includes lipids, that lipid bilayer. So something without a membrane doesn't have that lipid encapsulation. It's just
right
not there. Okay, so I just learned. This is the first time I had really seen this. So now this is how we learn. And I'll tell you what, when we read peer-reviewed articles like this, sometimes it is so cutting edge you won't see it in your textbook. The textbooks are like 5-10 years behind what's printed. And so sometimes the only place to learn it is actually in the article itself, and it tries to explain what the term is. So you can see how difficult it is to write a textbook, because this is what you have to work with. You don't have a textbook to help you get through. You have to be the one to write from there. Okay, so typically involving RNA metabolism, MLOs, these membraneless organelles, serve to concentrate their components in a way that facilitates processes such as transcription. So can you explain for the viewers what transcription is?
It's when the DNA is like being like kind of copied as RNA, like maybe I don't know the whole process really, but okay.
Close enough. It's when RNA is RNA uses DNA. An RNA is made based on a DNA template. There are probably better ways to say it than I just did.
Okay, wait. Let me try to describe the process. The DNA gets like split open, and then it connects to the RNA, and then the RNA like goes over and to the ribosome. Am I getting this?
You're getting pretty close to it.
Fix me. Fix it.
Well, you know what? Actually, now I'm a little embarrassed because there's something with polymerases and all that. And the nice thing is you can actually just do a simple Google and say DNA transcription, and it'll be like a 3-minute video, and you'll see it. I mean, if you want to do it, we can do it now, but we might get a copyright hit. Okay, so
no. Let's go on. I don't have to understand that whole process to get what's going on in the article. Go ahead.
And it says ribosomal RNA, and I'm embarrassed now because I should know this. And rather than just pull it off the top of my head, we could Google. Can you Google ribosomal RNA and show it? No, I'm glad we're going through this process because I'm showing you an ordinary individual without a lot of biology background can actually study a paper like this if they're willing to just kind of quote unquote have a dictionary or an electronic textbook, which is Google, to help you figure out the terms. And this is how I learned a lot of stuff because, like I said, some of this isn't in the textbook. So even when Dr. Sanford sent me off to school, he would be having me read papers like this, and I had to figure out what this was.
Wow. Okay.
And you can see that it's okay. Let's say it just takes me 10 minutes to figure this out. The next time I see it in another peer-reviewed paper, it's not going to be intimidating because I'll have some idea what it was. So if you go through 10 hours of this, if I could just give a little pitch here on behalf of doing things like this. Now I know, Rebekah, you and I were on your channel for 7.5 hours this past week. I can almost guarantee you, if you invest the same amount of time just once in nerd torture like this, you'll be talking up there with the best of them. I mean, you'll be so fluent, and they'll say, if one of them says, evolutionary theory is so good, and you'll say, well, they made a failed prediction about junk DNA, and then you can read off this paper to them. And they'll say, do you understand? And I said, well, I understand parts. I understand it enough to know that you guys are wrong. But anyway, so what is a ribosomal RNA? I actually could use...
you want me to read the definition? Okay,
yeah,
here we go.
If you can uncoil it.
Yeah. It says ribosomal nucleic acid rRNA is the RNA component of ribosomes, the molecular machines that catalyze protein synthesis. Ribosomal RNA constitute over 60% of the ribosome by weight and are crucial for all its functions, from binding mRNA and recruiting tRNA to catalyzing the formation of a peptide bond between two amino acids. Even the structure of a ribosome is determined by the three-dimensional shape of its rRNA core. Proteins present in the ribosome serve to stabilize the structure through the interactions with the core.
That's good enough. Okay, so let me explain this a little bit. So it turns out, most of the time they tell you that proteins are the machines of life. In this case, RNA can actually be also part of that machine. So you have a machine that's made of both proteins and RNA, and that is just too cool. And the RNA that happens to be part of this machine, and I love the word machine, and I know that's going to get under someone's skin, so I just love saying it. The ribosome is a machine, despite what some of my detractors refuse to call it a machine. It's made of both proteins and RNAs, and an rRNA is the kind of RNA that ends up in the ribosome machine. So there we go. Thank you. See, now I can be unembarrassed now that I've learned that. And Paleologos.
Now, look, but I have to say this. Look, I can sit for 7 hours on one of those type of streams, okay? But this nerd torture, I mean, we've only been doing this for an hour, and I'm just already like at my limit. I'm just like, I've been sitting in this chair too long. I'm like getting fidgety, and Yeah,
Okay.
I might be getting close to the limit of the
Okay. No, this is
the sciencey terms I can handle.
no, this is fine. Actually, this is telling me you're really learning. You're creating new neural pathways in your head because it's taking so much effort. And I'm embarrassed to say it's been so, you know, I focused on so many other things. I needed a refresher. What you just did actually helped me. That an rRNA is a ribosomal RNA. I've had so many different classes of RNA in my head: link RNA, LNC RNA, miRNA, and I just like, I just please stop. All these classes, and the rRNA was a, and you're just going to get so many of these. There's another one. There are all these funny names of RNAs too. Pee-wee RNAs. I mean, and you have these scientists that specialize in just one class. And they all just end with RNA, but then they put all the little prefixes. Okay, so maybe since we're there, we can stop. We can stop because
Okay.
the next is up. Yes. So the next
I appreciate this, Sal. I'm very happy with this, but I'm also glad that we're gonna slow it down.
No, because
I'm not earning my nerd badge tonight. I didn't earn my nerd badge tonight.
Well, I'll tell you something, and this is just from my experience too. I knew when I was studying biochemistry, after the first 7 minutes, I couldn't learn anymore. It's like, okay, if someone told you to memorize a Bible verse, it's like, okay, I'll memorize a few words. If they just try to force you to memorize a whole chapter in one sitting, it's just, unless you have a phenomenal memory, that's very hard. You have to do it in small pieces. So this is, I'm glad you're being honest about where you are and how much you can take. And this is about the right amount, because I got to tell you, when I took my biochemistry classes, I was totally a waste after the first 7 minutes. It's like, how did I get through it? Well, I had to go home and actually spend 11 hours reading it in small pieces. And so your experience is not at all unique. Oh, and guess who's here?
Otangelo's here.
Yeah. Oh boy, I have another
person. Tag team. We can tag team.
Tag team. Now can torture someone else. But do you feel like do you feel now that you can say what they call junk DNA wasn't? You have good evidence. You can cite this article, and if they say you don't know what you're talking about, it's like, okay, all right, maybe I don't. Why don't you explain to me why this shows that this? Why don't you explain to me why this article shows that RNA and that the genome is mostly junk? Because when I am reading all this sounds like it's very well designed. I mean, you know, it can really irritate. It's like, can you do better? Can you do better? Could you build a cell from scratch that does all this? You know,
even if we gave you all the parts.
Even if we gave all the parts.
Even if you had all the parts, like Dr. Tour says, I'll give you all the parts.
I'll give you all the parts. So, Otangelo, would you like to join? I sent you a link privately. One reason I don't post the links publicly is we can get imposters who will, you know, bomb the channel with obscenity, and we don't need to go into detail. So, but I'm really glad, because this also gave an opportunity to review all the stuff that we had on your channel about eukaryotic components. We reviewed chromatin, and we reviewed the central dogma of molecular biology. See? And you even knew the words like transcription, and you were able to describe the role of chromatin. And I quizzed you on I said, what kind of cell is this? Is this a eukaryote and prokaryote? Identified it. See? You're ahead of most creationists in being able to defend the creationist viewpoint. And of course, most creationists...
Well, thank you for helping me, Sal.
Yeah, most creationists will run circles around me because I don't know I know nothing about paleontology and all the layers and the names of all the fossils. I don't focus on that. And anyway, Otangelo, would you like to come in? I'll give you a link. It's in your email. So feel free to jump in. Maybe I'll just shut this down if you don't want to come in. Could you say something, Otangelo, if you want to come in? I'll wait a little bit. Okay, why don't we decompress a little bit, Rebekah? Talk about some random things.
What would you like to do?
For a couple minutes for Otangelo. Okay, he said just a minute. All right.
Isn't Paleo Logos coming on your channel? Is he the one coming on Tuesday when you do the genetic entropy? Oh, good, good. And so for those, I mean, I know there's only like 2 people watching right now, and this channel is just getting started. But if you happen to be watching this channel and you don't know Paleo Logos, please do go subscribe to his channel. And Otangelo has several channels. One of them is called The God Talk. So you can subscribe to that as well.
Let me go run in.
He didn't get your email.
I'm going to resend it. 1 second.
Okay, sure. Sal's leaving me alone with his audience. I can say whatever I want. Just kidding. So, wow, this is crazy. I have no idea what I should say. Oh. Paleo Logos. Oh, it says, have you seen Jackson Wheat's short section on you in his book? Sal, I guess you maybe you're featured in Jackson Wheat's book. Maybe I'll have to read that. That'd be interesting. All right, everyone. Well, I think Sal is just leaving me alone, and I have nothing to say on this channel, guys. Sal, hurry back. I'm like this. I'm trying to fill up dead space, but I'm not being very entertaining.
Oh my goodness, you're actually at a loss of words. I can't believe it.
If I was on my channel, I would find something to say, but I'm on your academic channel.
Look who else is here. Oh my goodness.
Oh, wow. Lindsay's here. Oh, wow. Oh my goodness.
She
was
saw
watching my show last night.
Oh boy,
Oh, wow. Okay, well, I will. Hi, Otangelo.
Yes, hello, Rebekah. Hi, Sal.
Hi.
Oh,
hi.
And by the way, Lindsay, Secure the Harvest here is having a stream tomorrow at 6 a.m. my time. I guess it's 7 a.m. your time, or her time. It's 8 a.m. your time, Sal and Otangelo. But she's doing great work on a slightly different topic. Like she's doing a lot of like biblical history and archaeology, really delving into, the evidence. What's up?
I'm going to make Lindsay a moderator. And the reason I'm doing that, Lindsay, is you can type, if you want to add the link to your channel and also the link to that show, you can do it now since I just made you a moderator. So I'm trying to help get you some free advertisement here and people can find it. And Otangelo, I'll do the same for you, sir. You can advertise your work here on my channel. Thank you for visiting.
Yeah, thank you.
So
and Lindsay did correct my advertisement. Her thing is going to be on Monday, not tomorrow morning. That's good because I didn't get a lot of sleep, but I was going to wake up for her thing. But I'm glad I can sleep in past 6.
Bravo, a Monday morning stream. Oh my goodness. Bravo. What a way to start the week. So I made Paleo Logos a moderator too. So
cool.
See, it's great if for the club that's here, and you guys can feel free to just provide links to your YouTube channels and advertise, self-promote. That's quite all right. So Rebekah, I hope it's been beneficial to you.
Yes, thank you very
You could
much
hold
for
your head
having me
high.
on. What?
And I said you could hold your head high and say, "Why are you listening to Sal?" And you could say, "Well, all Sal did was just read from a peer-reviewed paper. What do you have a problem with?"
Yep. Okay, thanks for having me on, Sal. Bye, Otangelo. Bye,
Thanks for
everyone.
joining.
Bye, Rebekah.
Take care. Otangelo, long time, brother. God bless you.
Yeah,
Man,
God bless you.
I don't think you've been on one of my streams in like forever. So
Yeah, it's a long time that I'm not joining you to have some review about some stuff, right?
yeah, it's been a while. I've had, like for the month of March and April, I was really focusing on the physics stuff, which hardly not even Emory wanted to be around me when I was doing that, because I had an electrical engineer and that was about it in Benrec 777 when we were doing that. And then prior to that, I was involved. I had Rebekah and then also Cindy Lincoln as my co-host, and so it's really I'm glad to be reunited. Um, Part of it was because Emory was a graduate student, and it was really good to have a graduate student in biology reading stuff with me. But I said I feel kind of bad because I promised that you and I could have a show sometime, and we finally got around to it. And I think this is a topic you'll like. So, just to spool you up on, I don't know if you had a chance to read the paper. We were just reading it for the first time, even for myself, right on air. I mean, I didn't go from top to finish before this. I said, this is why I do the shows. It's just so I could read it and also do it on the air at the same time. And
Right.
yeah, just like when you and I did the stuff on the irreducible nature of eukaryotes. That was a great show.
Yeah.
Well, what's on your mind, brother? What would you like to do with the remaining time? If you had anything on your heart, you had the floor.
Yeah, I was previous to join your stream at Brazilian Atheist channel, and I'll give you the link here quick. They were enjoying this science paper from Nature, claiming that an RNA self-replicating molecule was finally proven to exist, and that it was published in this article at Nature magazine, 18th of March this year. Evolutionary transition from a single RNA replicator to multiple replicator network. But what they use here is replicase. Let me check here where they say. Here we perform a long-term evolution experiment of RNA that replicates using a self-encoded RNA replicase. So I had asked them, well, were there replicases on the prebiotic Earth which helped RNA to self-replicate? And where did you get RNAs from? And they were biologists, professional biologists. They said, well, RNAs, they were just around on the prebiotic Earth. And they said, heck no. that is just not true. There were no RNAs just on the prebiotic soup. Nobody knows how you can have RNA prebiotically.
And oh, by the way, I'm going to have a Fazal Rana. I interviewed him, and I just haven't published it on my channel because it was a private Zoom link. And I told him, I said, if anything needs to be edited out, I'll do that because he's such an important person. And he was saying, yeah, the RNAs have a half like cytosine, like in a hydrothermal vent, would be gone in 19 days. Half of it would be gone. I was like, that's like no time.
Yeah, I said that as well. I said ribose has in 40 days it's gone. It is very fragile and
I didn't know that.
disintegrates. Yeah, it disintegrates very rapidly. And RNA general, all the bases and so forth, they have a very short half time. So
yeah, one thing that Fuzz Rana emphasized, he said, yeah, all those experiments are irrelevant to abiogenesis.
absolutely,
And of course, so this is what's very dishonest about the whole industry. They'll make all these experiments and say we proved something and it's not relevant. They'll pretend it is.
well, I questioned them tonight, and they just say you are using sources which are not credible. Like I cited Stephen Banner and people like that, and I quoted them and said, "Hey, these materials." Someone came in and said, "Well, but these prebiotic soup molecules, they had billions and billions of interactions, and somehow something positive came out of it." And I said, "Well," so I took Stephen Banner's Paradoxes in the Origin of Life and quoted him, "Hey, these molecules, they disintegrate and become asphalt."
Yeah. You see, this whole industry is able to push forward because people are willing to be dishonest.
Yeah.
It's not that they have the facts they don't have textbook facts. It's exactly as Fuzz Rana said, where's the relevance? As James Tour pointed out the same thing, as he said, it's dishonest, it's misleading. At best, it's delusional. He didn't use the word delusional. I'm using that word. At best, it's delusional. And I'll say at worst, it's fraudulent. It's either delusional or fraudulent, somewhere on that spectrum. It's anything but realistic.
Has Rob Stadler given you a link to a paper about fine-tuning of phospholipids? Have you heard about that?
Who?
Rob Stadler.
Oh, Rob Stadler. He may have. I have so many things to be sorting out through right now in my life. I'm going through kind of a big upheaval. I prefer not to talk about what's going on. It's mostly good, but it's wearing me out. And so this is, strange as it sounds, it's actually relaxing to be talking about this stuff. So by the way, you have moderator privileges. You don't have to drop this in the private chat, Otangelo. You can put it in the public chat.
Right, I'll do that. Yeah, I'll do that.
Yeah, thank you, Otangelo. And that way it's recorded for people that have time and interest. I think I have interest. I don't always have the time to follow up. So tonight if you're at all curious, we can change the topic at this point because Rebekah kind of said she was at her limit for what she could digest tonight. And I said, I totally get that, because when you're in learning mode, you're kind of inputting and reprogramming your brain to learn new things. And that does take effort and energy. So if you want to talk about the paper we talked about today, we can. Otherwise, I'm open to changing the topic, since we've been a long time since we've seen each other. And we could probably do that for about another half hour. And then probably I'll have to check out, because I'm running out of gas myself. You might see me on another stream, but when I'm running the stream, it takes a lot of energy out of me. So What would you like to do? Do you want me to explain what the paper was tonight?
Whatever you want to talk about, I'm fine, Sal. So
okay, yeah. Well, maybe this will help you. And do you want to put a link to your channels in the side chat? Why don't you tell me about your main channel, and why don't you introduce yourself to the audience while I grab something to drink? Would that be okay?
Yeah, sure. I'll put here in the side chat the link to my.
Yeah. So I just need to take a little bathroom break and get a drink. Sure. So why don't you put your channel and what you do?
Yeah, I'll do that.
I just want to say, guys, Otangelo is a great guy, and I'm really glad to be reunited with him tonight. And he and I are finally partners on a show after a very long time away from each other. So Otangelo, take it away, and I'll be back in a few minutes.
Sure. Okay.
Oh, Otangelo, you didn't have to wait for me to come back. I was hoping you'd say something.
Yeah, I just put the links to my YouTube channels and my library in the side chat, so anyone can. And I was reading the article which you sent me to chat about the role of RNA in biological phase separations.
The reason this is important is 10 years ago, this Dan Grauer, evolutionary biologist, was just saying if ENCODE is right, evolution is wrong. And
If ENCODE is right, okay.
yeah, and are you familiar with the ENCODE project of the National Institutes of Health? It studied chromatin modifications. One level of, I mean, it was one of the first big initiative to study chromatin modifications, the level of RNAs that were transcribed. So we know 98% of the DNA doesn't code.
Right.
So what does the rest of the RNA, what happens then?
MicroRNAs, which have regulatory functions, and they are part of gene regulatory networks. They regulate things, right?
Well, microRNAs are maybe less than 1%, and then we have ribosomal RNAs, and then we have a lot of these other ones like intronic RNAs, and then.
But ribosomal RNAs, they are part of the coded section, right? Or not?
No, they're not. So this is amazing because you actually have RNAs that don't code for anything that end up part of the ribosome. We just read it that 60% of the ribosome is made of ribosomal RNAs.
Oh, that's interesting. So that means it is transcribed to become RNA, and that is then immediately ribosomal RNA, which then will fold to become the ribosome. Is that correct?
It's part of the ribosome. 60% is, at least according to the article Rebekah just read. And I felt a little embarrassed because my focus, when I took classes on RNA, it was on link RNAs and microRNAs. I didn't actually study ribosomal RNAs, what they call rRNAs.
Yes.
So I was a little embarrassed, and I'm glad I had Rebekah just kind of read. And so we're going through the paper today and just identifying the classes. And there are all these other ones like peewee RNAs and snow RNAs. And I'm like, okay, I'm getting lost in all of these because there's so many classes now.
Yeah, the snow RNAs, they also are, they have also business in making ribosomes. And you have the nucleolus, which is the factory of the ribosomes. And there you have other compartments and organelles which are also employed in making machines that make ribosome RNAs. So you have basically machines which make the subunits of the ribosome. That's incredible.
It is.
And once you have that product, then you have error check and repair mechanisms which check if the product is okay, and then it goes into the assembly of the ribosome. And if it is not okay, then it is either recycled, the RNAs, or it is discarded. So, of course, you need to pre-program. You need to know what is the right situation, what is the wrong situation, and then you can make the error check if it's okay or not, the product. And of course, you need to know all this, right? And
yeah, and I'm so swamped with the stuff that I study. I'm more on the protein side. I'm not an RNA guy. And even if you're a protein guy, usually a protein researcher will focus on only one protein, like say topoisomerase, and that's enough to fill out several professional lifetimes. Just one protein, because it's so difficult to understand and carry out all the experiments to figure out what each piece does. And it's only 1,500, 1,600 residues, 1,600 amino acids. And you got people spending their lifetimes studying what it does. I mean, in that time, I could probably memorize the entire sequence by heart, but it doesn't mean.
But you know, what I don't see is many people which are actually studying systems biology, where you try to have an overview and try to understand the connections of the different parts of the cell and how they work together. And
It's
that's
because
basically, yeah.
I would just say the reason is I think it's because it's too complicated. We can only get little pieces. And there's a joke, would a biologist be able to fix a radio? Because a radio is a systems level problem. Biologists are still at, kind of the piecemeal, just fragments of the puzzle. So I'm sorry, go ahead.
Yeah, I mean, I agree with you. I mean, if you want to have just an understanding of the ribosome, it is so complex. And as you said, you can study it an entire lifetime. And there are people which are studying it for 40, 50 years. And they have still so many things which are not understood. And I mean, it's just crazy.
And the funny thing is, even though we don't understand it, the evolutionary biologists are confident that evolution explains it.
Yeah, I mean, that's their starting point and their faith, which they think explains everything. But the more that we actually study, the more we know that's not true.
So this whole thing, so let me just give you some proportions. And Dr. Dan, in my debate with him, gave all the proportions of the classes of DNA. And ENCODE verified that most of all of the DNA is transcribed at some point. So the introns are transcribed, and we don't, it's only now we're finding out how one of the ways that they're used. This paper covers how relates to maybe how it's used. So it's a big deal.
I don't know. So that how up to what point you have studied fine tuning in biology. I started to understand that Watson-Crick base pair, the hydrogen bond, which joins the base pairs and which permits the Watson-Crick ladder, that force is finely tuned. And that fine tuning, that force depends on the isomeric configuration of the atoms of the nucleobases. And they are just right in order to get the right force so that the base pairing can occur. So that was my first thing which I recognized in regards of fine tuning in biochemistry. And as I said previously, Rob Stoddard, he sent me a paper which I put here in the side chat about fine tuning of phospholipids. And I mean, it is amazing because if you think that things must be fine tuned, that means you have a state of affairs where things don't work. But if they are finely adjusted and tuned, then you have the function which is desired. And obviously that means that if you want to explain that through evolution, you have an entire evolutionary process and a state of affairs which is in a transition. But these transition zones, they are not functional because they are not you are not getting there to the fine-tuned situation. So evolution doesn't explain fine-tuning in biochemistry. It is an all or nothing business. And I think this is very powerful evidence which goes against evolution and favors design.
And I'm so glad that you don't get interrupted to say that, because if you're on another channel that will not be named, you'll have Leo Filius interrupting you rudely. You're not even able to finish your sentence and point out.
Yeah, when I see him, then I don't join the channels anymore.
It's just not worth your time. You see, you're teaching me.
Right. Yeah.
You see, I would value hearing that from you. Leo is just in such attack mode, he won't let you, and he'll just ridicule you and not try to understand. And I'm just like, okay, you can wallow in your willful blindness here, or you can try to consider it.
Yeah, I mean, that's why you don't see me probably so often. I mean, I had recently great opportunities to go to different channels. I was in one atheist channel in India. Today I spoke on a Spanish channel. It's fun. I am my wife bought Netflix, and there are some great series in Spanish. So I'm looking soap operas in Spanish. And Spanish is very similar to Portuguese. And
You speak Portuguese.
yeah, I mean, I live in Brazil, so
Yeah.
you know. And this afternoon, I was on a Spanish channel speaking in Spanish, and I had great fun. You know, and
That is, and you can speak Spanish too. That's great.
yeah.
You can speak
So
Italian. Can you speak Italian?
yes, my parents are Italian. I have
Yeah,
passport Italian.
You're a citizen of Switzerland. Can you speak German?
Yeah, I have double passport. I have a Swiss passport and an Italian passport.
But
Since
you can speak in German, can't you?
I grew up in German part of Switzerland, so that's my
So
first
you
language
speak
actually.
Italian, you speak English, you speak German, you speak Portuguese, and then now you can speak Spanish. God
Yeah,
bless
right.
you.
Yeah,
Praise God.
yeah, yeah, yeah.
Well, this is a chance for me to compliment you, sir, because you've taken a lot of flack from the other side. I mean, you can expect that they're going to say all sorts of vile things about us because we're Christians and we're creationists. Even if you're an
Yeah,
accomplished individual like you obviously are, that's just how it goes. Um, by the way, I highlighted the fact Secure the Harvest. Her first name is Lindsay. She's subscribed. So thank you. So perhaps you could return the favor. She needs some subscribers. So let me see. Lindsay, if you're still there, feel free to post your link again and maybe you can get some subscribers. We call that cross-pollination in the YouTube world. We subscribe to each other's channels just to help our subscriber base. So going back to the paper Rebekah and I were covering. So Dan Grauer said, if ENCODE is right, evolution is wrong. And so what ENCODE discovered was that practically all the DNA was transcribed. They used that to make their claim that therefore at least 80% of the DNA is functional. And
right.
the other side, I think, rightly claimed, and I said I actually sided with them, that that could mean that it's just transcribed and it's still junk. And I said, guys, they do have a point here. They do have a point here, the evolutionists. I said, just be a little cautious and just be patient because we might discover the function of the RNA. Because at the time, 10 years ago when ENCODE made that announcement, so guys, please subscribe to Lindsay's channel if you haven't yet. I already have. She's been helping me understand all the mechanics of trying to make YouTube shows. So she's been very generous with her time to help me. So anyway, back to the topic. At the time, they said, this is evidence it's functional, but they weren't able to actually get into the details. And so the evolutionists were having a field day saying, they're just, in fact, the language that the evolutionists use, they said those researchers are ignoramuses. They're crooks. Their leader, Ewan Burney, is the scientific equivalent of Saddam Hussein. And the reason they were saying that, particularly Dan Grauer, they said if ENCODE is right that most of the genome is functional. If ENCODE is right, evolution is wrong. If ENCODE is right, evolution is wrong. So the creationists took that and ran with it. They said, well, ENCODE is right and evolution, therefore evolution's wrong. I was more circumspect. I said, I think that they're right. And I'm pretty sure evolution is wrong, but we need a little bit more research to prove their claim. Emory's here. Emory, I'm going to make you a moderator, brother. And if you want to jump in, you're welcome to. I sent you an email. and I just made you a moderator. So you could search your emails, and you should be able to jump in. We're about to close. You can drop in, and we can go bash Dan Grauer, who said, if ENCODE is right, then evolution is false. And so tonight was kind of the finishing move. If you ever watch some of these video games, when the victor finally finishes off his opponent, this is the finishing move. We're finding all this function for these RNAs now that are transcribed. And this is devastating because they staked their whole thing on saying that the genome is mostly an accident, 90%, somewhere around that figure. I hesitate to, they keep changing the figure because we keep finding more and more function. And it's like, well, what are you going to have left at this rate, to say that it's junk? Now, I do believe...
So this thing that most of the so-called junk DNA has function, this is known already for several years, right? And I mean, those which simply want to believe in evolution, there is no evidence which will change their mind. They will just stick to it because that's what they want to be true.
So I just want to say, Emory, you're welcome to join in if you want. We're just shooting the breeze now, just talking a little bit about the paper. We went into the abstract in the first paragraph. Oh, he's here. Hey, brother.
Hey, I apologize. I sent a message to my family earlier today saying, hey, can I call? Because my dad usually works on Sundays, and tomorrow's Father's Day. So I wanted to call, and they called me like 10 minutes before the show started. I'm like, hey, I'm like, okay, fine. That's not going to happen. So sorry about that all.
Well, happy Father's Day to your dad, and you're a good son, sir. He should be very proud of you. So we're kind of almost just wrapping it up. We had just me and Rebekah, and so that was, that the direction of the show was going to be very different if it's just me and Rebekah versus me and you.
Yeah. Yes, it would be a lot less nerd.
And so we just got through the title, we got through the abstract, we got through half of the first introductory paragraph in an hour, and that was still very good because I said, Rebekah, let's look up all the terms. Do you remember all the stuff that we covered on examining evolution? And I said, if you spend 7 hours of this, you'll really know a lot more than most creationists, and you'll be able to hold your own against a lot of evolutionists.
Mm-hmm. Yeah.
And I said, Rebekah, you had like a 7.5 hour stream recently. She said, no, but that's different. She said, after an hour of this, she's already kind of worn out because you're trying to input and learn all these terms. And I said, I hope you feel better. You accomplished something. You can hold your own. And so that's all we did. In other words, if you just read the abstract in half the first introductory paragraph or the first paragraph and a half, you will have caught up to everything we did. But it was still a very good thing. It was satisfying to see her kind of invest time to learn new concepts.
Right.
And this is why I have an academic channel. On my main channel, that might not fly for people who want to get entertained. So,
right, yeah.
So this is the academic channel. So anyway, we were talking, she said, I didn't know this whole thing about if ENCODE is right, then evolution is wrong.
Dan Grauer.
Dan Grauer. And you know what? I'm just, the evolutionists now are kind of embarrassed. You won't hear them argue that junk DNA is junk hardly anymore.
Not too often.
Yeah,
I mean,
because
I used Grauer's textbook on genetics in my very first graduate school class. First class I ever took was was a Dan Grauer textbook.
Oh wow!
And it was funny because he was just ripping encode in the text, and my teacher was kind of like, "Yeah, I don't necessarily agree with that." Like, yeah.
Hey, please. I know, like you and me, we have to start. We have to make decisions for what books we collect and keep. Can you please keep that textbook? We need to.
Yes, I think I still have that textbook, so I should be able to dig it up and let you know what textbook that is.
So if you're thinking about doing a fire sale on that, at least say it for me. I'll pay you the fire sale price. I might not be the, if it gets too expensive, it may not be worth it. But I almost want to to quote what Sandy Pigeon said, one of my friends. I think you met him.
Yeah.
We ought to frame it. And it's like, can you evolutionary biologists get anything right of major importance?
No. The answer is no.
No.
I mean, I suppose they do occasionally stumble onto something. Even a broken clock's right twice a day. But
yeah, that's right. Like on the show where I broke all these papers on RNA phase separation, I also said, just for kicks, I threw in the fact that they'd done a study on synonymous mutations and found most of them are harmful.
Oh, I saw that. Yes.
And Dan was saying, oh, that's only 5% of the genome. And I'm saying, okay, that's still half of the problem for, you only need 1% to be functional. And that's beyond Mueller's limit. Evolution could be wrong. But the devastating point is, look at all the papers that assume that synonymous mutations are neutral.
Yeah, that just blows neutral theory right out of the water. That was my first thought when I saw the title. I'm like, oh, that's the end of neutral theory right there.
Thousands of papers are wrong now.
That's like 90% of population genetics papers too will assume neutral theory. Like, do they realize how much of their dogma they're going to have to blow up to fit that paper?
I just like, yeah. So even though, I was like, yeah, I feel for Dr. Dan. He's a good guy. In my book, I mean, as far as his dealings with me, he has friction with other people. He calls Nathaniel Jeanson a liar and all these nasty things. At least he's nice to me. Okay. So, he'll have to settle his fight with Nathaniel Jeanson. I'm not going to get in the middle of that. Obviously, I side with Nathaniel. But I had to feel for Dr. Dan. I said, Dr. Dan, this does not look good for your field. This looks really bad. And if to only make matters worse, we have these papers on RNA phase separation. And now we know what introns do. Now we know what all those transcribed RNAs ENCODE was talking about. They can be used to create these membraneless organelles. And it naturally self-organizes, and it's, by the way, it's sequence dependent. So the sequence.
So much for the introns being spots for evolution to occur because their sequences didn't matter. There goes that.
I mean, it's not so dependent. I mean, okay, so you could compromise a nucleotide here or there, and it's just going to end up in a slightly different location. It may not be a total deal breaker, but enough of that accumulates, it's going to break something. And so they might be able to isolate some of these few experiments where they'll knock a few pieces out and it still works. And it's like, guys, that's a terrible metric, because if it happens to have deep levels of redundancy, you're not going to detect the functionality with single knockout experiments.
Yeah, and I mean, the single knockout experiment to me, where they say, well, look, we can get this one. Oh, okay, but that's not, you don't seem to understand the scale of the problem here. This is like the Titanic having an iceberg taking a huge chunk out of the hull. You got water gushing in like crazy, and the ship's carpenter's running around next to the porthole, and he's nailing a board over it. Said, "I got this one." You're taking on water. You're sinking, and you're busy nailing a porthole shut.
So
that doesn't solve your problem, guys.
So, I put this on the main channel just in passing, and people loved it. And, I said I could get, I'll get the usual treatment. Said, Sal doesn't understand the paper. It's like, I think I understand a lot more than you do. And the more important thing is the paper's the paper. Mm-hmm. People ask me, how do I find this stuff? I happen to be I'm well connected to people in secular science, because there are some of us that are creationists or ID proponents that work in secular universities. Some of them are professors whose name shall not be named. And I will just I'll get something in the email and say, hey, like for this one, for the Ka-Ks ratios, Ka being the number of synonymous mutations or non-synonymous. Okay, Ka is non-synonymous. Ks is synonymous. I don't know why they didn't use Kn, but that's okay. And they've been using that in population genetics forever. It was Joe DeWeese who sent me that one. And then the stuff about phase separation, I can't name those names. I can name Joe DeWeese. I got to hear them, and they said, yeah, we work across the hall from guys that study this stuff. And the question was circulated, it's like, well, do they believe it's junk? And he said, none of us believe it's junk. And so
then tell Dan Grauer.
Yeah, go tell Dan Grauer. And I said, okay, so the researchers that actually do stuff like this, that conduct the experiments, they don't think it's junk. And the more important thing is they're making a good case, both with their experiments and then their colleagues with all these papers that are published that describe it. So I specifically started then to Google sequence dependence phase separation. And then, oh my goodness, the spatial organization of all these ribonucleoparticles that are membraneless organelles throughout the cell is dictated by the sequence of the RNA. And I'll tell you, because the sequence of the RNA, it's not by itself. There is associated protein binders, RNA binding proteins that will target that sequence. So collectively, the complex then, you have to evolve the protein, obviously. Then you have the RNA. The combined effect ends up locating it, but it's a little bit more than that. The RNA has methylations on it.
Right, yeah.
And that's what they call the epitranscriptome, the collection of all the methyl modifications.
Right.
And then they have A to I edits, adenosine to inosine.
Right.
They're all so yeah, the methylations, A to I edits, and then they've identified like 60 different modifications on RNA. 60. That will modulate the behavior of the ribonucleo complex. So you could, not only do you have kind of the base location, but then you can modulate it with these RNA modifications and the variety of them. And I think, guys, this is a lot more complex. I think you evolutionary biologists are kind of trivializing all this. I think this is a lot more functional and complex than you ever dreamed. And I was just celebrating. I said, on top of that, we have do you remember our flip-on talk like 3 months ago?
Yeah.
CRISPR-Cas wanted to get on and just poo-poo it. And he said, oh, that's only one example. I'm like, dude, maybe that's the only one we discovered so far. Are you so sure? And he said, oh, yeah, I'm sure. Because if it were if it were functional, he would have found it by now. I'm just like, dude, I'm not going to get in your face, but I think that's one of the stupidest things I've ever heard.
That's not that.
Sal, it's late and I'll go to bed.
Oh, Otangelo, thank you for joining. So we did get Emory and Otangelo and Rebekah, everyone we were hoping.
Yeah. So let's keep in touch and see if we can do other streams and review some science papers and
Yep.
have some fun.
Yeah, maybe it'll be just you and me, because I know if it's me and Emory, oh my goodness, you'll see how it turns. Well, you just saw how it's going to be if it's me and Emory.
Yeah.
I'm sorry, Otangelo.
No problem. It's all right. So.
Well, God bless you, brother. You take care.
Yeah, God bless. Have a good night. Bye-bye, guys.
Yeah, Otangelo, take care, man.
And Emory, if you want to advertise your channel, you can do that too.
Yeah, I mean, I'm not really doing too much right now. I have 2 videos planned. Actually, make that 3. I have 3 videos planned, but so I thought I was done with my graduate research, and then my professor decided that he wanted me to go mine a bunch more data. So I'm currently busy mining my way through 10,000 species of insects.
Oh,
yeah. So, and that's not even all I have to mine through, but I have to finish that by the end of the month. So,
okay, this is, guys, viewers, this is life as a graduate student.
Yes, this is life as a graduate student. And I'm such a masochist. I'm thinking about getting a PhD.
Yeah. You know what? I specifically asked on your behalf.
Mm-hmm.
I said, Dr. Joel Duff, I said, do you have a problem? Okay. If you saw my 2-hour show with him and you didn't figure out that I was really asking on your behalf, I said, Dr. Joel, would you have any problem having a young Earth creationist be your PhD student? And he said, no, of course, unless he has an attitude. Okay, so I went to bat for you there, Emory.
I appreciate it. I think Dr. Duff and I would probably kill one another, so I don't think that's probably the best option.
All right. Okay, so maybe it's the thought that counts.
It's definitely the thought. I appreciate you asking, Sal. I appreciate you asking.
It's like, okay, you told me you were looking for a place. I said, okay, I know at least someone, since he directs a PhD program, and I didn't mention you by name, but you're the one that was on my mind. I just want to let you know I was going to bat for you there.
I appreciate it. I appreciate it. So backstage, I'll tell you something that you will find hilarious. Yes, so I've been looking at programs, and I found something that I think you will find very, very funny, but I'm going to say it backstage rather than out in front of everybody.
I understand. Yeah, we need to both be careful for all sorts of reasons. Okay, I'm glad you got a kick out of me asking Dr. Duff on your behalf.
That is actually legitimately funny. And I'm actually somewhat surprised he said yes. Although I'm not. I want to preface this by saying I'm not questioning Dr. Duff's integrity, but I could see certain evolutionists saying yes and then using it to destroy the person.
Yeah, well, he said, okay, now if he came here and said his goal was to destroy evolutionary theory, he said he would have a problem with that. And that was honest, okay?
Yeah.
I felt that was, and I get where he's coming from, because there are definitely some areas where you wouldn't use evolutionary biology and you're a young Earth creationist. So like if you're like, trying to help develop vaccines or something or whatever, it's like, well, yeah, maybe you don't really need to believe in universal common ancestry. But he said if we had a young Earth creationist come in there and said, I'm here to learn this so I could disprove evolution, he said I'd probably have a problem with that. And I thought that was, I appreciated him just being forthright and honest. And, I'm like, okay, you're just telling it the way it is. And, and that's perfectly fine. And I've had some professors who are ardent evolutionists, and they kind of give the same answer. And I think that's fair if it doesn't affect your field. I mean, if you're a cellular biologist looking at cells now,
I don't think that really has that much play.
Right. So I thought that was, I've heard the same from others. So just to know, I'm glad I told you that just so I could get a laugh out of you there, Emory.
Well, I got a chuckle out of that. That reminds me of another funny story that I'll have to tell you afterwards as well. So,
So where was I? So I felt bad for Dr. Dan because he was on the junk DNA train, and I'm just like, it's not looking good.
Yeah, that train is headed for a cliff, and I really hope everybody gets off before it crashes.
That's why I'm glad I hope you keep your textbook from Dan Grauer.
I think I still have it. So I'll have to find it because I've got like 3 bookshelves worth of books. But I think I still have that, so I'll have to dig it out.
And I've been digging it, yeah.
And I'll try to remember, because you know where I'm going to see you. I will try to remember to bring it when I see you, so I can show it.
Okay. Yeah, we could talk about that in another place. Let's not.
Yeah.
And so, and the other thing is, I have to dig up the population genetics book to talk about the Ka-Ks ratios.
Yeah, I probably should get my hands on a population genetics textbook. I don't think I own one, so I probably should have to get my hands on one of those.
You can get one free.
Or is that Felsenstein has his online?
Yeah, that's the brutal one. And
okay.
You know what? If it makes you feel better, I don't like population genetics anyway. I have the math skills, but I'm just like, this is kind of ugly math to me, and it's pointless anyway. I'm sorry, Dr. Felsenstein, if you're listening. I think our show.
Some of the simplifying assumptions are really, really bad.
Well, the funny thing is, Joe Felsenstein said that. He said the one Lucas models approximations are, they're bad.
Yeah, they're not very
good. And then the killer is their definition of fitness. Like Washington said, the most fit organisms are the ones that reproduce the most, and the ones that reproduce the most are the most fit, or something like that.
Completely circular.
Yeah, it was bad, and I'm like, it's not very helpful. So actually, it's damaging. I mean, they spent 100 years on this, and it's like you guys haven't figured that out. That this is useless. It's taking me this long.
That is the equivalent of what one of our fellow YouTubers, who's a atheist, came at me when I helpfully defined woman for a certain person on Twitter who couldn't define it. Read Supreme Court Justice. I defined woman for them, and this individual came back at me and didn't like my definition. I said, "Okay, what's your definition?" And this individual responded, "A person who identifies as a woman." And I said, "No, you don't get to do that. That's a circle. That's a giant circle. A woman cannot be a person who identifies as a woman, because then the term is meaningless."
I didn't realize that. That's pretty good.
So, and it's the same way. It's the same way with fitness. You cannot be the most fit if you produce the most offspring, but you cannot make. But at the same time, be defined as the most fit by producing the most offspring. Like, you can't. You're circling yourself into the middle. You can't do that.
Right. And we covered that for 10 hours with R.H. Brady, and then 4 hours with, kind of my upcoming book. And then, so, by the way, guys, I should do a little bit of shameless self-promotion.
Please.
This gang, minus Otangelo plus Paleo Logos, we're having a show on genetic entropy. Oh, before I forget, if my computer dies in the middle of this, I have a backup. So just hang on in case we cut off. Let's see, I have it here. So let me, okay, let's see. So that's shameless plug for this coming Tuesday. I call it Genetic Entropy 2.0. That's not the title of the book. The title of the book most likely is going to be The Destiny of the Human Race. And part of that will be kind of a simplified and also augmented genetic entropy. The main difference between the way that I frame it versus the way Dr. Sanford framed it in 2005, and I think he's sympathetic to some elements of the way that I'll frame it, is the definition of fitness. And this all proceeds from that paper by Lenski that says the genome decays despite sustained fitness gains. I'm just like, uh-oh. I mean, this is because, go on.
Wow. Please tell me in that book that you are quoting Dumber and Dumber. Please tell me you're quoting that.
What should I quote from Dumber and Dumber?
No. That particular sentence where it might be our destiny to get dumber and dumber.
Oh, yeah, that's him.
Please tell me you're quoting that.
Okay. Oh, sure. I'll say, please go to our website, and we'll you can see.
I'm sorry. Go ahead.
To save the human race, we all get dumber and dumber till we all lose our minds. Oh, I got you now. Oh my goodness! And you know what was really funny about that? The guy who said that didn't even realize he proved my point so brutally. It's like, I was trying to teach you something. That was the whole point of me showing you R.H. Brady's paper. You couldn't have proven my point than what you just said, and you still don't see it. And that just kind of makes it funnier that you don't see it.
Yeah, just
pathological blindness.
Spiritual blindness.
Yes. So why don't I read you the abstract of the paper, and then we can just shoot the breeze a little bit more before we sign off.
Sure.
Let me see if I can. I mean, so it's been a good week for the design proponents.
Every week's a good week when you're right.
Yes. And, I mean, and I feel for Dr. Dan because he's been very friendly to me. And, he and I did stake out our positions in a debate that's recorded and archived for all posterity. And, in light of this, it's like, I think the weight of the evidence is showing that we're right.
Yep.
Dr. Sanford and I, but I got to tell you, and I took flack for this because I think it was only like a year and a half ago, I came on Dr. Dan's channel and he said, I want to talk to you about ENCODE. And I said, Dr. Dan, I think I agree to this extent with you that RNA just being transcribed doesn't mean that it's functional. And I said, I think I would like to caution my fellow creationists not to use that ENCODE quote. We're not there yet. Just wait a little bit. And I got an earful from SFT trying to lecture me. And of course, that was kind of the beginning of the end with me and him. But I hung around. I said, it's going to happen. It's going to happen. I just sense it. And then only like this month, someone said, phase transition, sequence to phase transition. I'm just like, whoa, I think I know what you mean. And these are guys that are working on this. And I just happened to be lucky. And just that, okay, so this was like an hour and a half talk. And in just 10 seconds, I got what I needed. The word phase transit. And that was all that was mentioned. It's like, oh my goodness, this is it. We're here. And this is in addition to my flip-on talk. I mean, these are polyfunctional systems. And I already added a little bit with the ALU elements.
Mm-hmm.
Now that's only 10% of the human genome.
Right.
But I talked about the adenosine to inosine editing. and at least, you only need 1% of the genome to be functional for evolution to be falsified for as far as humans. And, I'm just like, well, we bumped that up to at least 10, we added another 10% just on that. And that wasn't me. Francisco Ayala ended up having real egg on his face, and I just couldn't help but gloat. Because he's saying that's an incompetent designer who'd make ALU elements. I'm just like, yeah, right.
Mm-hmm. Totally incompetent. Totally.
So now, then we had flip-ons, and then, oh my. Oh yes, SFT is planning to stream with Kent Hovind to debunk me about geological column. Laugh out loud. Oh my goodness. I don't watch. I think I can only take so much torture. So anyway, why don't I read the title in the abstract? Yep. We'll talk about more wholesome things and
yeah.
That other. So it says, oh, by the way, Paleologos, I got your back here. Even though I know that you have some disagreements with the characterization my boss had about contested bones and all, I view you as on the same team, and it's cool. It's okay to have disagreements with you and with me. All right, I'm not gonna attack your character or anything, or your skills or your competence if we disagree. It's just part of the process. And I've changed my mind on a lot of things too. So I disagree. The present Salvador disagrees with the old Salvador, or the younger Salvador. So we're cool, and I hope you know that. Oh, thank you. Yeah, I want you to know I consider you a friend, and I hope you'll feel free to be a colleague, and if we can mutually support each other, that would be great. So anyway, I'm just going to read through the abstract, and then we'll just go from there. The role of RNA in biological phase separations. And this was actually in 2018, 4 years ago, and I can't believe I missed this. So it's been around a while. But only recently we started to get a plethora of papers on this. And I was Googling around, and I thought this was only in 2021 or 2022, but it actually has been around for a while, and it's just been quietly progressing. Marta Fay and Paul Anderson. So Division of Rheumatology, Immunology, and Allergy, Brigham and Women's Hospital, Boston, Massachusetts. And Paul Anderson, Department of Medicine, Harvard Medical School, Boston, Massachusetts. Yeah, you wouldn't get this from an evolutionary biologist. Phase transitions that alter the physical, so the abstract. Phase transitions that alter the physical state of ribonucleoprotein particles contribute to the spatial and temporal organization of the densely packed intracellular environment. This allows cells to organize biologically coupled processes as well as respond to environmental stimuli. RNA plays a key role in phase separation events that modulate various aspects of RNA metabolism. Here we review the role RNA plays in ribonucleoprotein phase separations. In summer, I think the reason I found this article, I was looking for sequence-dependent phase separation. So the sequence will you see the position of all the RNAs throughout the cell? The sequence will determine its positioning in 3D space and in time. And there are all these classes of RNAs that I needed to learn. Like, I wasn't really that familiar with rRNA. I don't know anything about SNO RNAs. And then they have these snRNAs. I know some link RNAs like hot air and NIST, but then there are these other ones, NEAT1 and MALAT1. That's your organization. Do you want to read this for me? Can you read it?
I can. You're breaking up pretty badly. Is there a link somewhere I can grab?
It's in the video description.
Okay, it's in the video description. Okay.
Yeah,
Okay,
when I
there it is.
scroll, it breaks up because my computer can't handle it.
All right, fair enough.
Oh, Paleologos, while Emory's digging that up, do you believe in the geological column? I think Jim Clary does.
I think most creationists outside of Hovendites do.
Yeah, I think it. I mean, you have a column there. You have different strata. I mean,
Yeah, that's a whole kettle of fish there, I'd imagine. But
yeah. Okay, I'm not. I think it's really obvious my lack of geological and paleontological sophistication. I'm a cell biology, protein biology kind of guy. I'm not. I'm not that kind of creationist.
All right, so yeah, I stay out of rocks. The only rocks I am interested in are the ones in my head, and even those I'm not that interested in.
Yeah.
So I guess if you handed me a dinosaur fossil, I'd be interested in that too. But anyway, so the abstract, oh my goodness, holy, this is either really long or really short. One of the
It's
two.
a very short abstract, and then the next paragraph is organization.
All right, so phase transitions that alter the physical state of ribonucleoprotein particles, that's a big word, contribute to the spatial and temporal organization of the densely packed intracellular environment. So it organizes the inside of the cell, essentially. This allows cells to organize biologically coupled processes as well as respond to environmental stimuli. RNA plays a key role in phase separation events that modulate various aspects of RNA metabolism. Here, we review the role that RNA plays in ribonucleoprotein phase separations. All right, and then there's a graphical abstract with a bunch of different types of RNAs and where they're used. I don't know all of these RNAs. I know some of them.
Yeah, I don't know them all either.
Short and easy translation. There's a bunch of different types of RNAs that do a bunch of different things.
Yeah.
There's a very short, simple explanation for you. I could tell you what some of them do. I'm not familiar with all of them, though. I'm not a cell biologist, really. It's not really my thing. So let's see. Do you want me to start reading the organization section, the next section?
Oh, please.
Okay. Organization of the densely packed intracellular environment requires compartmentalization. This is particularly important for gene expression as coordinated processes must occur in an ordered fashion. In eukaryotic cells, double-stranded DNA, dsDNA, is sequestered in the nucleus and packaged in histones. Within the nucleus, DNA is organized into heterochromatin and euchromatin to control the relative access to the transcriptional machinery. Transcribed mRNA undergoes splicing, polyadenation, and capping prior to export to the cytoplasm. So basically, just make that make sense. When the cell makes RNA, mRNA, messenger RNA, it does a couple things to it before it ever sends it out into the cytoplasm to be translated. It does some work with it before it ever goes out. So you're not just, I know some of you may have gotten high school biology and thought, oh, DNA makes RNA, which then makes protein. Well, yes, but it's a little bit more complicated than that. There's a lot of things that happen in between those steps. Mm-hmm. Each of these processes is under spatio-temporal control that ensures correct processing and localization. Just as membrane-enclosed organelles, e.g. nuclei, mitochondria, endoplasmic reticulum, Golgi apparatus, serve to organize biological processes into discrete cellular domains, non-membrane enclosed domains play a similar role in the organization of biological activities throughout the cell. These are defined by the physical nature of their constituents and are referred to as membraneless organelles, or MLOs.
I learned something right there. I did not know that. It's not in any of my textbooks.
Yeah, that's new to me too. I'd not heard that phrase MLO. due to their ability to concentrate factors associated with a biological process, typically involving RNA metabolism. MLOs serve to concentrate their components in a way that facilitates processes such as transcription and ribosomal RNA, rRNA, processing in the nucleolus or sequestration, such as translation initiation complexes and signaling molecules in stress granules, SGs. MLOs, which, reminder, membraneless organelles, can be visualized using phase contrast, immunofluorescence, or bright field microscopy. You don't need to know that. The lack of a limiting membrane allows MLO constituents to rapidly exchange with the surrounding environment, allowing a dynamic response to changes in the cell state. So let me translate that for you. Basically, because these organelles don't have membranes, they can react quickly to changes in the cell. Right? So
Yeah.
that's the translation of that.
And we have that diagram here. I'm showing it on the screen.
Okay,
And you
fantastic.
can, isn't that
Yeah, so
awesome?
Yeah, I mean, let's look at it this way. There is a lot of cool stuff going on in that diagram, and I'm not even sure what all of it does.
I've heard the term perispeckles a lot, and it says the proposed function is modulate gene expression by sequestering protein components, microRNA production. Oh, this is complicated. These perispeckles.
Yeah, complicated doesn't even begin to explain it. Good night.
And we have these first time I heard the word cagal bodies and nuclear speckles. I've heard nucleos before. And these stress granules. First time I heard that.
Stress-specific mRNA storage.
Whoa.
Well, wait, hold on. If I'm understanding that correctly, what that means is that there are ways in the cytoplasm for the mRNA to be stored for a later time.
Mm-hmm.
Folks, I don't think you maybe realize how big of a deal that is. RNA is highly unstable. Like, highly unstable. Like, I'm not commenting on the goodness or badness of the COVID shots, but there's a reason that a lot of them have to be stored at incredibly low temperatures, because otherwise the RNA that's in them just falls apart. How do we know how long it can store that?
I don't know. So this is all new to me.
Yeah, I'm gonna have to look that up and find out how long they can those stress granules can store mRNA, because wow.
But we have to commend the researchers that can figure this out. This is brutal research.
Wow. Okay, I'm. This is too late at night to be learning this much stuff. Man, this is cool. Now I want to go down a research. Now I want to go down a research rabbit hole, and I have to actually go to bed at some point tonight.
And I just, even when Rebekah couldn't understand, well, no one really understands this unless you're like in that.
Unless you have a PhD in cell biology and this is your field.
Right. But both she and I, as we read, even just reading some of the abstracts, she said, and even like Mark O'Connell, he said, they're still insisting this is junk because they're describing. I'm just like, well, that would at least give me pause not to just come out and say it's definitely junk because of evolutionary theory. I said, you ought to be a little bit more cautious, because
a little more circumspect here. Good night.
I mean, and I got to tell you something that's looking really embarrassing for Dan Grauer. When he wrote that thing about the Gospel of Encode, the immortal.
Yes, I remember that. Yeah,
he had like 5, 6, 7 co-authors.
Mm-hmm.
His latest stuff, it's solo authored, and a lot of it's just on archive. It doesn't even make it to a peer-reviewed journal. It's bad.
That sounds like some of his colleagues are starting to back away from his ideas.
Yeah, do you want to be associated with that? Do you want to come out with
if I'm a rising evolutionary biologist or even a rising geneticist, and I'm thinking to myself, man, I want to have a nice long-term career here, and it looks like junk DNA may be going the way of the dodo bird, I may not want to co-author a paper with Dan Grauer.
Dan, And guess who one of them was? And I've actually corresponded with him, Richard Acevedo.
Huh.
And he was nice to me personally, but I think he was just like a flamethrower like Dan Grauer.
Yeah.
And I was kind of surprised to find out he gets appointed to be the chair of the biochemistry department. And I was thinking, yeah, okay, so you're the chair of the biochemistry department. You want to get grants on cell biology. Do you think going out there and saying the RNA's junk is going to help you get grants and fill out the positions of your faculty?
Yeah.
So we don't see him tooting that horn anymore.
Yeah, that's
So Dan Grauer is going to be in a pretty lonely position. People are like, "Hey, Dan,
yeah.
sorry, buddy, but the NIH is offering lots of nice grants on RNA that you're saying is junk. You really want me to sign on to that anymore?" Sorry, I can't help it, Claude Emory.
Yeah, and I mean, science is about who's providing the funding, to be perfectly honest. So if the NIH is now funding, oh, RNA has a function. Oh, this section of the DNA has a function. Oh, this section of the DNA has a function. If they're funding that research, Dan Grauer is going to be in a lonely cubicle very, very quickly.
Right. Okay. I got this in a discussion with a professor. Do you know what the golden rule is?
Follow the money.
It's close. He who has the gold rules.
Oh. Oh my.
So praise the Lord that the Lord has caused the need to solve medical problems, and that's where the gold is. And it's forcing the scientific enterprise to actually deal with the data rather than to keep subscribing to a dogma like evolutionary biology that doesn't work. That doesn't help us understand how biology works. And so God is giving us a lot. He's given us, I saw this way back in 2016, 6 years ago. He's given us an ally that we didn't expect. We creationists.
Yep.
And so, yeah, like you said, Dan Grauer sitting alone cubicle.
Yeah, I mean, and I don't necessarily fault, by the way, I want to make this clear. I don't necessarily fault Dr. Grauer for sticking with his hypothesis because he's invested a lot of time and energy and effort into this hypothesis. It's hard for someone who's put that much time into it to abandon it. But at the same time, Dr. Grauer, I don't think he'll ever hear this, but you may want to, for the sake of both your career and your integrity, decide, you know what, I'm going to own it. I'm going to eat some humble pie and figure it out. But most professors don't like to do that.
Yeah, I don't know, and I'm just sorry. Maybe, this is the one time you see maybe not Sal in not so nice mode. I'm really gloating now. I'm sorry, because I remember what it was like in 2012, and I was getting browbeaten by Larry Moran and Arthur Hunt and Steve Matheson. And they were just, huh?
I said, I know who all those people are, and I know exactly how this conversation went.
Yeah. And not only was I a creationist, I didn't have any biology background, and then we didn't have all this. And they're browbeating me. And it's just like, guys, it's payback time. It's payback time. And, I can't help but gloat. It's like, yeah, I told you guys so. And I was just a lowly creationist. Doesn't it kind of hurt now? Oh, and Matheson's another story. He's now editor of Cell Reports, editor-in-chief. I'm like, yeah, you better start to change your tune. You want your journal to stay alive, right?
Yeah.
Because you're going to miss out on great articles that cover stuff like this.
Yeah.
So,
and Cell Report is a fairly popular journal, so they'll probably be okay for a while. But if they hang on to that bin for too long, they could be in some trouble.
I'm sure he's probably changed this tune by now.
I would imagine.
You're not going to stay editor-in-chief for long. if you
Probably not.
if you don't deal. The only way you'd stay editor-in-chief to keep spouting nonsense, evolutionary nonsense, if you're editor-in-chief of an evolutionary biology journal. You could stay there forever.
Yeah, pretty much. Yeah, unfortunately.
Would you like to read the figure and then maybe just one more paragraph, and then we'll just
Sure, we can read the figure and then we can read another paragraph. No problem. No problem. Let me pull it back up. So the figure, it's a figure, for those of you who are just listening, it's a figure of the cell, and it has, I think, 6 different markings on it. So I'll read each marking, and then I'll read what the proposed function for that marking is. The first thing is the nucleus.
I'm going to show the diagram here. Okay.
Okay.
There's
fantastic.
a diagram, guys.
Yeah. So the first thing I'm going to look at is the nucleolus, which underneath is rRNA and snow RNA, which I'm not sure what snow RNA is. Sal, do you know what that is off the top of your head?
No.
Okay. So the nucleolus,
small nucleolus or something. Go on.
Small nuclear organelle RNA or something. So the nucleolus is the rRNA processing and RNA modification place. So that's apparently, according to what they're proposing here, they're saying that basically when RNAs are made, they're sent to the nucleolus for modification and processing. Next thing down is what's called the paraspeckles, which have the adenosine to inosine edited RNAs and the LNC RNAs -neat1. I'm not sure what the neat1 stands for. You know, so?
I think that's the name of the link RNA. And so the link RNAs have all these funny names like hot air and NIST. And so that's another one.
Okay.
I think it's a specific link RNA.
All right, so the paraspeckles are supposedly supposed to modulate gene expression by sequestering protein components and produce microRNA. Sequestering protein components? Okay.
Okay.
Okay, I'm going to move on because otherwise I'm going to say something that I want to say at the end of this.
Yeah.
Nuclear speckles, which is looks like long non-coding RNA, MALAT1, and then poly-A RNAs. Nuclear speckles, they are supposed to be involved in mRNA. What's RNA? I'm not familiar with that.
mRNAs?
Yeah.
Those are messenger RNAs.
No. mRNA. It could be a typo.
Yeah.
I'm guessing that's a typo because I maybe unless somebody knows something different, but I'm going to go with that being a typo, which would be mRNA maturation, including regulating splicing and mRNA export. Yeah, I think that's a typo.
Yeah.
I think that's a typo. So apparently the nuclear speckles are involved in regulating the splicing of the RNA. So after the RNA is made, it splices to make the protein component that the cell wants. And it also helps with the export of the RNA into the cytoplasm. The caudal bodies, I'm not sure how to pronounce that correctly, which works with snow RNAs and snRNAs.
I don't know what snRNA is either. So you have SNO and SN. So you have all these other ones.
Yeah. Then you have that's RNA modification, processing, and assembly of small RNA complexes. So maybe small nucleotide RNA, maybe? I don't know. But so it's involved in RNA modifications, processing, and assembling of small RNA complexes. Then there's the processing bodies, which are mRNA exclusive, and that's mRNA storage.
So the processing bodies.
More storage?
Yeah.
Okay. And then there's the stress granules, which we already talked about, which have the poly-A mRNAs and have stress-specific mRNA storage. Evolve this. Okay.
I wonder if these are all MLOs, these membraneless organelles.
They're within the cellular. They're within the nucleus, and the nucleus is a membrane-bound organism. So let's say they're within the not necessarily. They might be all MLOs. Yeah.
So MLOs in the cytoplasm and also inside the nucleus. That would be. I mean, I'd have to read.
Yeah, I'd have to read a lot more literature on this.
MLOs.
Yeah,
And,
I'd have to read a lot more literature here, but
yeah,
jumping.
this is a good day for kind of just spiking the ball on Dan Grauer's head. I'm sorry, and Larry Moran, and Arthur Hunt. Oh, Arthur Hunt, I just despise now. He'd make so many personal attacks on me now. The more education I got, the nastier he got with me. And Arthur Hunt, if you're reading this, there's a saying in the Proverbs in the Bible: correct the fool and he'll hate you for it. Correct a wise man and he'll love you. And that's helping me understand our dynamic and our relationship, Arthur Hunt. Must not feel good to be corrected by me, right, Arthur? Sorry, I can't help it. Rub it in. I mean, I was totally nice to him as much as I could take it, but that was started to go over the top when he started to take real personal shots at me consistently. I'm like, dude, that's beneath you, and I didn't deserve that because I've been nothing but civil to you. And, I could only take so much of that. So anyway, you can read the next paragraph then, and then we'll just talk about something else, and we'll wrap up the show.
Holy jumping. Oh, my. Okay, let me read the next paragraph.
Oh, so those are MLOs. It just says figure 1, MLOs.
Yeah, it does, yeah. Membraneless organelles.
Wow.
Holy smoke.
This is new. I was telling Rebekah, I said, these aren't in the textbooks. How do you think the textbooks are made? The people will read the papers and then make it a textbook.
Yeah. Oh, my. Oh, my. Oh, my. Okay. I'll read the next paragraph, and then I'm going to call it, I think. But
yeah, me too. So, I'll meet you backstage for a little bit afterward.
Yep. MLOs are assembled via a process of phase separation. During phase separation, a critical concentration of a given component, e.g. protein, RNA, ribonucleoprotein complex, allows a transition to a concentrated phase which is physically separated from a more dilute phase. So basically this looks to be a gradient of concentration.
Yeah, which is really cool because it can leverage things like Archimedes' principle or whatever. They'll just kind of, well, I'm jumping ahead here. Just go ahead.
Yeah, no, but you're right. Yeah. So this process is driven by multiple weak electrostatic charge, and repetitive domain interactions that demix a homogeneous state into two discrete phases. Sal, you know more about this than I do. Explain that.
Okay, remember they were saying all these repetitions are accidents. You have a lot of repetitive elements in the RNA and DNA. Now you're seeing the importance of it because it changes the physical. I've been telling Dan this. I said it has biophysical properties like structure. And I was kind of close to the mark, but this is really, I mean, I think definitely in DNA, but in RNA, this is saying that the number of repetitions of your motif is actually important to the way it's going to behave. It really means something,
Yeah.
You can't. So your transposons that are like, you have the microsatellite repeats and then these transposons, it may or may not add up, but they're detecting when it does add up to something significant. They can detect it. Because if you have a repetition, it changes the charge configuration.
So basically what you're saying, Sal, is that DNA does physics.
Yes, and the RNA. There are two things. It's functional in the DNA state, the repetitions. It's really functional in the RNA state too. And this takes you're making it polyfunctional. And I've seen this in other things too that are just mind-boggling, like in the zinc finger protein. The protein is optimized to work with the DNA and then also the RNA transcripts by two separate mechanisms. I mean, the amount of foresight that's needed to be able to make this Rube Goldberg machine just boggles the mind. And it was so sophisticated, even the researcher said transcription factor 3 alpha, which is a zinc finger protein, said it's sophisticated. I'm like, that's kind of an understatement. It's like fearfully and wonderfully made by creators, more like it. I mean,
yeah. This thing did not fall into place by random chance processes. I'm sorry.
Yeah.
No way.
So this is some biophysics here. This is some serious biophysics. This is not, multiple weak electrostatic. These are very, these are just kind of the positive and negative charges. And they're just right. And so when they say charge, this would be like positive, negative, or positive, or it's repulsive. And
it would have to be positive because otherwise, or negative because otherwise it would attract.
Right. So this, yeah. See, this is not trivial to be able to make a membraneless organelle
and make it all stay together.
Right, because you're gonna
and be able to come out of that state when you want to.
Right. And so now let's explain a little bit about the repetitive nature of this. So you have this, for lack of a better shape, let's just assume a sphere, like physicists want to do. So if you want to have all certain charges on the outside, you'd probably have kind of the same nucleotides represented on the outside versus the ones on the inside. So there's, it would have to repeat, across the sphere to give it that, certain charge configuration. So there's foresight in how all this RNA will also fold with the protein. I mean, this is like non-trivial. The amount of foresight to conceive of this, it's just, and you know what? To an evolutionary biologist, it just looks like a dumb repetition. It's like, that's because you're not a biophysicist,
Yeah. Okay, next sentence. This process can produce a discrete liquid state within a liquid known as liquid phase separation, or a transition of the portion of the liquid into a solid state, causing a solid within the liquid.
Yeah, this is good biophysics, guys.
Yeah.
Do you know any chemists that can conceive of stuff like this? I mean, this is like non-trivial chemistry and biophysics.
Yeah. Oh my. Let's see here. Molecules within a phase-separated liquid domain, which typically assumes a spherical shape, can continuously exchange with the surrounding solution while maintaining phase separation.
That's pretty incredible. This is like a membrane-bound organelle, but without a membrane.
So let's take a step back and think about that for a second. You have a liquid that is within another liquid, and it's phase separated by charge. Keep in mind, this is by negative, or positive charge. But it can exchange pieces across the charge gradient. Wow.
Yeah, wow. Wow is right. And my specialty is in condensed matter physics. This is mind-boggling, guys.
Folks, let me suggest an experiment for you sometime. Take two batteries, line them up, positive end to positive end, and see if you can get them to exchange. See how well that works for you. I got news for you. You do the same thing with two magnets. Line them up positive end to positive end. I got news for you. They repel each other.
Yeah.
You're not getting bits of one magnet going to the other magnet. But somehow, you can get pieces from one phase into the other despite them having the same charge. Ooh, okay. The physics of that is probably well beyond my understanding. I'm a biologist, and I'm not a cellular biologist at that. But, wow.
How about the last sentence, and then we'll wrap up.
This is different than a transition to a solid state whose shape is internally defined by the patterning of its components, which rarely exchange with the surrounding solution. Okay, so when you actually make a solid, you're not doing a whole lot of exchange.
Wow. We had some fun tonight.
And I learned a lot. My brain got stretched.
This is how nerds go clubbing on a Saturday night.
Yes, this is exactly what I like doing with my Saturday night. It's a lot more fun than anything else I could think of to do.
That's why you're a graduate student.
Probably, yes.
Well, I hope you had fun. Do you have any, I've been in the stream for 3 hours. If you have any closing thoughts.
No, wow. I'm just, my mind is completely blown. So I'm good with shutting her down.
Could you offer a prayer or a praise to our Lord and Savior?
Sure, no problem. Dear God, our Heavenly Father, I humbly come before you, praising and glorifying you for the day you've given to us. Thank you for allowing us to spend some time together tonight. Thank you for your word. Thank you for the blessing that it is. And thank you, Lord, for this this paper and the information that it contains. Thank you for just blowing my mind with the incredible design that you've put into the cell, and the incredible way that the cell is able to process and store DNA and RNA. It's mind-boggling, the thing that you have done. And Lord, I pray that you will just bless us as we go to bed tonight. Help us to wake refreshed and ready to serve you in the morning. Help us to make it out to church in the morning. And Lord, I pray also that you will grant that the message will be uplifting and will be encouraging and challenging. And Lord, I pray that you will just also help us to accomplish the things we need to accomplish over the next few weeks. And may everything we say and do bring you glory and honor. I ask this in the name of your Son, Jesus. Amen.
In Jesus' name, amen. And thank you to all the viewers. God bless you. And Emory, I'll meet you backstage for a little bit. God bless you all. Take care.